Okay, good morning. It's just past 10 a.m. Good morning, everybody. First of all, I would like to thank you, all of you, for taking the time to come and attend this symposium on pancreatic cancer, a symposium fully funded by the Fundación Areces. Before I tell you a little bit about the Fundación, I would like to thank Mr. Manuel Azcona and his team for all the work they have done to put together this meeting. They have done absolutely everything, from the trips, flights, hotels, and to allow us to be in this wonderful auditorium. So thank you, Manuel, and pass my thanks to your people. The Fundación Areces is one of the top private organizations supporting scientific research in Spain. I have had the honor to be part of this organization for longer than I would like to remember as a scientific advisor. Today we are honored by the presence of the president of the Scientific Advice Committee and Dr. Emilio Bouza and Dr. Jose Maria Medina, who I think has even been at the Fundación even longer than I have been here. So we are all-timers. We are all-timers for sure. So I'll just take 5 minutes of your time to tell you a little bit about the Fundación. The Fundación Ramón Areces was established near 50 years ago, in 1976, by Mr. Areces. I had the pleasure and the honor to meet him a long time ago, while I was still in the U.S. And then I, knowing he passed away a few years later, and then his nephew continue, and then the part of the family continue being the president, the leaders of this foundation. This foundation is the main shareholder of the El Corte Inglés. Anybody who's been in Spain knows what El Corte Inglés is. It's the biggest department store in Spain. Has 84 department stores in different cities. It has even 2 in Portugal, been built recently. Mr. Areces was in the clothing business. He worked first in Cuba. Cuba was part of Spain until the end of the 19th century. Then he came back to Spain and he bought a small clothing shop when they made suits for gentlemen. In those days, they were not what they have now in El Corte Inglés, lots of suits so you can choose colors, sizes, etc., etc. They were made, they were made especially, to order, okay, for each person. So that small shop with 7 people is now what is the El Corte Inglés, one of the main business organizations in Spain. Mr. Areces was always interested in scientific research. He was actually a very good friend of our Nobel Prize, Dr. Severo Ochoa, and he has always supported the Center for Molecular Biology that has the name Severo Ochoa, currently at the Autonomous University in Madrid. So he's always been very keen in supporting science, and right now they have 3, I would say probably 4 major programs. One of them is to provide funding for research projects. This is I don't know, I have it here, like 6 million euro in funding for research, competitive research project. That is probably the main activity. Jose Maria knows much better than I the numbers. And the other one is also very well known in Spain. It provides support for young Spaniards who want to work abroad during their postdoctoral training. Support funding for people who go to the US, to the UK, and well, now in the US, I don't know, they're allowed to be there, but until very recently, that was probably the main country where people went, the UK, Germany, France, etc. And for a number of years, they didn't support PhD, but now there is a new program, already been a few years, to support PhD students primarily in Spanish institutions. So, and the third activity is what we are, doing today. They support lectures. They usually take place in the afternoon, so more people can come. Late outside Spain will be in the evening. Spain is late afternoon and 7 p.m., I think, are most of them. And symposium, like the one we are celebrating today. So I think I think you will agree with me. Obviously, we are all biased that pancreatic cancer is now going through a golden age. The way at least I see it. For the first time, we are seeing the possibility of treating people with drugs other than cytotoxic drugs. It's amazing how pancreatic cancer is probably one of the cancers that we know more about it at the molecular and on the cell biology level. And yet, right now in a hospital, any hospital in Madrid, in Barcelona, in the rest of Spain, in the rest of the world probably, are still treated with gemcitabine, discovered in 1997, or with FOLFIRINOX, which you have to be in good shape to be able to resist the toxicity caused by this mixture of 4 compounds. So I think we are now at the dawn of seeing this as a past, and now for the next few years, we are going to see the final entry into the world of pancreatic cancer of more, compounds that are more selective and, as a consequence, less toxic. So with this, just want to finish since we have a very busy day ahead of us, and I think I'm the chair of the first session, so I will remain here, and it is my pleasure to introduce to you Meritxell Rovira. She's a professor at the University of Barcelona, and she is a group leader at the nivel, which actually I'm not sure she will tell us, because I get confused with the research centers and hospitals in Barcelona. So the title of her presentation is Shedding Light on Pancreas Regeneration and PDAC Cell of Origin with Single-Cell Sequencing. I think all of you got this little flyer in which you have one of these QRs to be able to read the abstracts and the program. So with this is Meritxell here. Yes, of course. Okay, so let me say hello. Okay, I'll come up here about 5 minutes, so I'll give you
Sure. So, Good. Well, first of all, thanks to Mariano and Carmen and the Fundación Areces to inviting me to talk about the work we are doing in the lab. And stay with me, I will be mainly talking about healthy pancreas first, before moving to pancreatic cancer, and how understanding a bit better a healthy tissue may help us understand tumor heterogeneity. So I think the audience doesn't need to know what the pancreas is, but as I'm the first talk, I think I'm allowed to talk about the pancreas first. And the pancreas is an endoderm-derived gland with an endocrine and an exocrine compartment with different functions. So the endocrine compartment is in charge of regulating blood glucose levels, and the exocrine compartment is in charge of producing digestive enzymes for us to digest what we eat. 95% of the pancreas is exocrine. This is going with delay, that it's mainly acinar cells, the ones that produce and secrete the enzymes, and ductal cells, that are these tubes that drive these secretions into the duodenum for us to digest what we eat. In my lab, we are interested in understanding ductal plasticity and ductal heterogeneity for several reasons. On one side, because we are also studying diabetes in the lab, and ductal cells have been proposed as a source of new beta cells, although it's a really controversial topic. But you can find endocrine cells embedded in ductal cells in all mammalian pancreatic tissues studied. The ductal cells in the adult are transcriptionally really similar to a progenitor, so they have, at least at the transcriptional level, some plasticity. Work done during my postdoctorate show that these ductal cells in zebrafish, in an animal model that does regenerate the pancreas, because mammals do not have a high capacity of regeneration of the pancreas. So studying a model where you can study regeneration, we show that ductal cells indeed are progenitors in the embryo and in the adult. And moreover, more recently, it has been shown that ductal cells are uniquely able to form organoids. And I don't know how familiar you are with organoids, but are these 3D structures that you can grow in culture, that they come from a progenitor or a stem cell, and they recapitulate some features of the tissue of origin, and they differentiate to some cell types of the tissue of origin. So being a bit more physiological than a regular 2D culture. And moreover, we are also in the lab interested on studying ductal plasticity as the possibility of these cells to giving rise to pancreatic cancer. So far, most of the people has looked at ducts as a homogeneous population, and although it is not really homogeneous, when you look at the ductal tree, you have centroacinar cells, that are the last cells touching the acinar unit. You have intercalated and interlobular ducts that are quite small like this, and then you have interlobular ducts and the main duct. And I hope you can see with these hematoxylin-eosin images that it doesn't look the same to be a centroacinar cell than a ductal cell in the main duct, but we did not have the tools to understand if they were indeed different, because the markers that we used to study ductal cells were homogeneously expressed in all of them. So to study the differences between different ductal populations and different sections of the ductal tree, we used an already available tool. It was a SOX9 EGFP mouse that marks all ductal cells in the pancreas, not only in the pancreas. So it's a back transgenic mouse done in 2003, so we didn't invent anything. And it's marking with GFP all cells in the mouse that express SOX9. And in the pancreas, these are ductal cells. So this, it's not working, it's okay. So this allows us to image the ductal tree and also to separate the fractions of the ductal tree by size, and so bigger ducts versus medium ducts versus smaller ducts, and interrogate if the plasticity was different between the different sections of the ductal epithelium. And I hope you can see these are organoid cultures that when we plate the exact same number of cells in organoid cultures, when we plate ductal cells coming from big ducts, the organoid formation capacity was much higher, so around 8 to 10%, so here in red. This is the quantification of the images. So therefore, these cells in the big ducts had more plasticity, at least in vitro. And to make it a long story short, we designed a differentiation protocol to understand if the different organoids that we were generating had the same capacity to differentiate to endocrine cells in order to have a cell therapy for diabetes. And in this case, I hope you can see, we have proved this by RNA, but also at the protein level, that when the organoids were differentiated coming from big ducts, we were able to differentiate these cells into insulin-producing cells, while it was not possible coming from medium ducts or small ducts. So the next question was, what is in these big ducts that is different? It's not just the size of the duct, so there have to be differences in the cell types. So obviously, in the single-cell RNA-seq era, we did single-cell, and we fractionated the ducts by size in order to then being able to coming back and knowing if we can pick up a population, we knew where it was coming from, either from big ducts, medium ducts, and small ducts. We sequenced over 20,000 cells. And to our surprise, we discovered an unforeseen level of heterogeneity in the ductal epithelium. It was clear that small ducts were clustering separately from medium and big ducts, so the cells were indeed really different. And we defined classical markers from ductal cells, and we started to see differences like UNCAT, it's a transcription factor expressed in ductal cells, but we indeed saw that it was expressed only in medium and big ducts, but not small ducts. And on the other side, CFTR, that is the receptor for cystic fibrosis, was mainly expressed in small ducts. Besides that, we identified 15 populations. It says 16, but indeed it's 15 because we have 2 acinar contaminants in our analysis. We defined specific markers for most of the populations, and we interrogated if they were indeed different between them. And when we look at gene ontology, I'm not going to bother you with the gene ontology of all the populations, but I'm pointing out at one of them that we are further characterizing in the lab. It's what we call the Wnt-responsive population. It's coming only from big ducts. It's exclusively in big ducts. And it was really interesting because gene ontology was showing terms related to ribosome biogenesis and translation. And in the last 10 years, it has been described that quiescent adult stem cells have a high ribosome biogenesis and low translation activity. And this is the features that we are seeing in this Wnt-responsive population. Moreover, when we looked at what they look like compared to other cells in our body, they were really closely related to intestinal stem cells. And moreover, when we look at the KEK pathway signalings, the first one that showed up was signaling pathway regulating pluripotency. So suggesting that this population had some stemness potential, at least in the pancreas. And this is just a comparison of our population with intestinal progenitors, and they were really similar, but they are not expressing LGR5. So they are similar to LGR5 positive cells, but they do not express LGR5. And Adrian, a PhD student, was in charge of looking into this population in the pancreas to find the population. It took him 6 months. We almost gave up, and one day he came with the eureka that we have it. So he cut the main duct of the pancreas by chance, and we identified that this population, the Wnt-responsive population, is located in what is called pancreatic ductal glands. And these pancreatic ductal glands were described in 2010 by Sarah Thayer as a potential stem cell niche in the pancreas. And they are only located in the main duct and more abundant the closer to the duodenum the duct is. Then we not only looked at this Wnt-responsive population, we looked at all the other populations by Maxima technology. It's a multiplexing technology that allows you to multiplex up to 100 antibodies. We did not multiplex 100, we multiplexed 18. We isolated the main duct separately to be able to see it. And you can see here the glands. These small patches are the pancreatic ductal glands, and this is a regular section of the pancreas on the bottom. And just to tell you that we had, to our surprise, we were expecting less correlation. We had 70% correlation between the protein data and the single-cell RNA-seq data. So when we saw a population in a small duct, it was in a small duct. And when we saw the populations in big ducts, it was in big ducts. So proving that the single-cell RNA-seq data gave us the right settings. And finally, we interrogated some of these populations at the organoid level to see their plasticity, and obviously, not surprising, LY6D is the marker we are using to isolate the Wnt-responsive population and is the winner in organoid formation. It has a higher capacity than LGR5 positive cells to form organoids, suggesting that still it's even more stem, at least in vitro. But against our belief, when we interrogated if these cells were able to differentiate to endocrine cells, they were not able to differentiate at least efficiently to endocrine cells. They were really efficient to give rise to acinar cells in our cultures. And we have another population that we are further investigating, but I'm not going to be talking about, that it's the FLR3 population here, that it's able to give rise to all endocrine cells in culture. Just to show you, as I'm going to be talking about the Wnt-responsive population a bit more, we proved that this population exists in the mouse. These are the glands that Adrian proved that they were expressing the Wnt-responsive population markers, but not only in mouse, we proved with human tissue that these glands do exist, and these markers are expressed not only in the pancreatic ductal glands, but also in the periliary glands that are located in the common bile duct. We have a mouse line now that we are using to investigate the role of this population, both in development, in diabetes settings, and in pancreatic cancer. I'm going to show you some of the data. And this is just a CRI line that was kindly given to us by Esther Baena, that is a researcher studying prostate, not pancreas. And just to show you that indeed we have a super high efficiency of labeling of the glands using this model. And we are using it to investigate also if these cells could be a source of pancreatic cancer. And I'm going to show you that in the last slides. But I show you everything. It was done in mouse, and now we are moving to human. We have moved to single-cell multiome, and this has been a collaboration with Eduard Montaña to get healthy human pancreas from cadaveric donors and isolate the ducts to do single-cell multiome, so single-cell RNA-seq and ATAC-seq from the same cell. And Paco Real has helped us a lot in the bioinformatic analysis with Jaime on his team, Joel on my team, and they are in the team of the isolation. I'm not going to show you all the data, but with multiome, we had the ability to look at gene expression, chromatin. We have done data integration, gene regulatory networks, cell-cell communication, transcription factor networks. We have compared the data with PDAC data, and we have done cross-species studies. We have done this in 8 human donors, in 8 pancreas. We have proof that the results match with mouse. So markers that are expressed in small ducts in mouse are expressed in small ducts in human, and vice versa, in big ducts or in small ducts. We have novel markers that we did not see in mouse, like a lot of long non-coding RNAs or antisense RNAs. And we have also novel markers that we did not see in mouse, like MUC5B, that is exclusively expressed in big ducts. And this population had indeed been presented by others or described by others, but in such a small amount, because if you do not isolate manually the big duct, you normally don't get it in your single-cell RNA-seq. So sometimes it is really important to know your prep of origin, to know if you get all the information you want at the end. So this MUC5B population has been presented by others like a side population of ductal cells, and indeed it's not a side population, it's a population that it's not a population, it's a marker that it's defined by big ducts. With all this data, we have proof single-cell RNA-seq data and attack data showing how chromatin opening is allowing the expression of all specific genes. So we have obviously insulin in beta cells, we have glucagon, and we have specific markers like CFTR that I was telling you before. It's only in small ducts, but lowly open in big ducts. We have defined classical markers. We have shown also, like others like Luca Tosti in 2021, that there are two acinar populations, one that is highly secreting and one that is slowly secreting enzymes. We have big ducts and small ducts and so on. And what is important is that with multiome data, we can also interrogate the role of enhancers in gene expression on every single population that we have. So we now can map the expression to a given enhancer to know how this expression is regulated in every given cell. And finally, we were interested in looking at ductal heterogeneity, of course, because it's our cell of interest. And to make a story short, we have defined 17 populations in human. We did 15 in mouse, so it's not completely the same in human. These populations are more or less represented in all the human pancreas that we have analyzed. And with the multiome data, what it's really nice is that we can look deeper and we can look at gene regulatory networks driving the expression and the fate of every given population. So I'm not going to bother you with how we have done it, but with gene expression and chromatin data, you can create cis-regulatory elements identification and transcription factor networks that allow you to identify the transcription factors that are driving that given cell fate. Just to show you some, this is the Wnt-responsive population. The LY6D population has a unique network of transcription factors regulating the expression of the transcriptome of these cells. And also, we believe it's kind of the chromatin of the cells is quite open and it's kind of ready to do everything like a progenitor. And then we have a population that we did not identify in mouse, that we call it lipid metabolism population, because expresses all the machinery at super high levels related to lipid metabolism. And indeed, it has a transcriptional gene regulatory network that it's really different than the others. And then goblet cells that we have identified and TAF cells that we have also identified between others. So this is giving us a layer of information that may allow us to understand how cells are being regulated. We have compared our data in human to the mouse data, cross-species comparison, and I have to tell you that what I can say is that we have about 50-50% correlation. So not all the populations that we have found in human correlate to the ones we have defined in mouse, but most of them do. And just to show you some, so we have a proliferative population in human that matches perfectly the profile in mouse. We have EMT population, chemokine secreting population that matches perfectly. We can say that small ducts here on the top in human look exactly like in mouse. We have EMT population that I'm going to be talking about that it's present in both. We have the Wnt-responsive population in both, and we have the Wnt-responsive population and the mucinous population as well. So although it's not 100%, we can say that there are really many similar populations and some are different. And I think this is something we need to understand between different species. Not everything is the same. And it's important when we want to compare mouse to human data, because sometimes it not match what we want to see. And also we compare our human data to fetal human pancreas to understand these progenitor populations. And what I can tell you is this is fetal data. This is not our data. It's publicly available data. And this is our multiome data. And I hope you can see these are fetal ducts, that fetal ducts are much more similar to big ducts in human than to small ducts in human. So this plasticity is more present in bigger ducts than in smaller ducts. Oops, voy para otro lado. So, and finally, with this multiome data that we had generated, we are also really interested in understanding mesenchymal heterogeneity, because ductal cells, especially when the ducts become bigger, are surrounded by mesenchymal cells. And this mesenchyme may also play a role in pancreatic cancer formation at the end. So we wanted to interrogate the heterogeneity in the mesenchymal cells that we have in our analysis. And to make a long story short, we defined 4 mesenchymal populations. 2 of them are more pericyte-like, and 2 of them are more fibroblast-like. And now we are doing cell chat analysis that I'm not going to show you for a matter of time to understand how receptor-ligand interaction may influence pancreatic cancer formation between mesenchymal cells and ductal cells. And to close with what is important to everyone in the audience, we are looking at ductal cells as cell of origin of pancreatic cancer. We are not the first ones to say that this is possible. Already a few years ago, 2 labs almost at the same time showed that both acinar cells and ductal cells can be cell of origin of pancreatic cancer, but when the tumor comes from ductal cells in mouse, the mice die much earlier than when the tumors come from acinar cells, although acinar cells are more prone to give rise to pancreatic cancer. That's something that I think is important. And also there are differences in tumor evolution or formation. If they come from acinar cells, they have to go to what is called acinar to ductal metaplasia and then preneoplastic lesions, panins, to then give rise to a full-grown tumor. In the case of tumors derived from ductal cells, there is no acinar to ductal metaplasia, because acinar cells don't have to become ductal cells. Ductal cells are already ductal cells, so this step is not needed, and there are not panning lesions. So we go straight from a ductal cell to a tumor. So there are no preneoplastic requirements to form a tumor. So knowing this, and having first our mouse data, we compare our data to publicly available data of PDAC tumors and healthy pancreas. And I hope you can see, so these are ductal cells in control pancreas, so healthy pancreas, and this is ductal cells in tumors, what they call ductal cell type 1 and ductal cell type 2. And when we compare the profile of our populations, the transcriptional profile of our populations to this data, what we observe is that the Wnt-responsive population, interferon and EMT, that are populations that we are further investigating, were highly expressed in ductal cells in the tumors, more than in the healthy tissue, suggesting that at least they may be a cell of origin of tumors in this model. Moreover, when we looked at TCGA data of survival of pancreatic cancer patients, if these patients express a transcriptional profile similar to the Wnt-responsive population, interferon or EMT, they had a poor survival compared to any other population. And finally, with Jaime from Paco Real Lab, with the human data, we have compared the transcriptional profile of our human multiome of the healthy pancreas to a more recent paper that defined 4 signatures to classify pancreatic cancer tumors between basal-like A, B, hybrid classical A and classical B. And just to highlight that when we compare the transcriptional profile of the ductal populations that we have defined in human, not to our surprise, LY6D population, that is the Wnt-responsive population that we really know now that give rise to pancreatic tumors, is transcriptionally almost identical to basal-like A type of tumors in humans. And in case of signature 1, that is classical A, this is correlating with the transcriptional profile that we have in mucinous secreting populations in the ductal cells. So we believe that the cell of origin matters in pancreatic cancer, and that ductal cells give more aggressive tumors. I did not show you, but they are more metastatic, and response to chemotherapy is worse. But however, although we know acinar cells and ductal cells can give rise to PDAC, we know that PDAC is highly heterogeneous, and it's not a bimodal system. So it's not either acinar or ductal. There has to be something else. So we now, what we are doing is that we have created new GEM models to investigate how cell of origin influences tumor heterogeneity. We have selected 3 of the populations I've been talking about: interferon population, EMT population, and the Wnt responsive population, to understand if these cells are more prone to give tumors, and if these tumors are different between each other. The good thing is that the human data correlates, so we have also EMT population in human, we have the Wnt responsive population in human, and we have the interferon population in human that we had called it gluconeogenesis population. And we have created all the mouse lines. It took us a year to have all these. To study this in mouse models, in GEM mouse models. We have the Wnt responsive, interferon, EMT, and a pan-ductal Cre driver flock together with p53 flocks and KRAS G12D. And we are now studying this in a lot more detail, in a timely manner, to understand at several levels how these tumors are forming, how different they are between them, and if this correlates to human survival, and if we can find possible therapies for every type of these tumors. And just to show you some of the data, we don't have enough to show you a lot of plots, but LY6D derived tumors are small, they take long, but they are highly metastatic. And this is a pancreatic tumor. We have long mets, and this population is also giving us ampullary tumors in our system, because these pancreatic ductal glands are highly abundant in the ampulla of water. And to close up, I hope I had convinced you that cell of origin matters, and that it's important to look at the origin of the tumor in a bit more detail. This is the team that did the work, and all the collaborators, also the clinical collaborators, because we are lucky enough to have a lot of human samples that I did not show you the data, but we have a lot of PDAC samples from patients, thanks to the organ donors, and obviously this would not have been possible with the funding sources, and this has been mainly funded by CRIS contra el Cáncer and La Caixa. And thank you.
Questions? Let me ask you a question. With your ductal models, there are 4 independent promoters where you get the tumors. Yes. Do these tumors go through the classical pan-ins and 1A, et cetera, as you know, with the acinar?
We have not seen pan-ins yet. So what we have been investigating more is it's close to the end point. So we are moving backwards now, because we did not know the time frame of the tumor formation with these new models. So now, can you hear me? Yeah. So now we are moving backwards, but so yo estoy microfoneada. So far we have not seen pan-ins so far, as in derived ones.
Okay. A very quick, please answer. Oh, there is a question. Bruno, is that?
Can you hear me? Yes. Okay. Thank you very much. Very nice talk. Very elegant. I see a lot coming up. Interferon response or interferon response. I assume it's type 1 interferon response.
Yes.
And do you have any idea why? Because could this be potentially viral mimicry? What advantage might that have for PDAC and also the origins?
Super nice question. I don't know the answer. So we have to look in depth into that, but we believe it could be the case of a viral infection, even in a healthy tissue, because the population was initially defined in healthy tissue. So we don't know, and we are looking into it, but I don't have the answer yet.
Okay, thank you very much, Meritxell. We have to finish. Before I introduce the next speaker, let me tell you that this symposium has been co-organized by me and by Carmen Guerra, which I haven't mentioned her. She's not feeling too well today, so I don't know whether she is still here. Still here, okay. Well, just to let you know that she has been the major player in organizing the scientific part of this meeting. So I hope she can stay for the whole day, and if not, she stays long enough. She is the person who supervises the pancreatic work in our lab. So I apologize for not mentioning her at the beginning of the symposium. Bruno? Okay. Bruno Sainz is at the Instituto de Investigaciones Biomédicas, right? IIB at the nearby at the Autonomous University of Madrid. Okay, your turn. I'll get up in 5 minutes.
Okay, thank you. Thank you, Mariano. Thank you very much, Carmen, as well, and also the La Fundación for this opportunity to come here today and speak a little bit about the research we do in the laboratory, give you little snapshots of what we are doing and trying to focus or approximate our research a little bit more to the clinic. So I talked a little bit about the preclinical models we have in the laboratory and where we are going. So obviously, as we heard this morning, where we are with pancreatic cancer is not where we would want to be, especially if we compare it to other types of tumors like breast and prostate cancer. And although this statistic was published before all the KRAS inhibitors came out, suggesting that pancreatic cancer would become the second leading cause of cancer death in the US, but only behind lung cancer in 2030. I think that might change. As Mariano mentioned this morning, we are in a very new decade or era for treating pancreatic cancer. But I think if we're going to make advancements, I think all of us will agree translational research is really extremely important here. Preclinical models, animal studies, human studies, translation to patients and translation to populations. This is a bit difficult to monitor, yes. But translational research is a key, amongst other things, bioinformatics, analysis, single-cell RNA-seq, which has given us a lot of information. But I think when we combine all these things, we're really going to advance our understanding. So in the laboratory, we work on many angles, anywhere from tumor biology and tumor microenvironment to omics, drug therapy, metabolism, precision medicine, trying to take this all together, put this all together and try to apply it in preclinical models and hopefully in the future take it to the clinic. At the level of tumor biology and the tumor microenvironment, we know that pancreatic tumors are extremely heterogeneous. I think we saw this this morning. We've learned this and we've appreciated this, and this may be one of the reasons why it's very difficult to treat pancreatic cancer. The extremely heterogeneous really make it a one-size-fits-all approach, quite impossible for treating these tumors. We believe that the cancer stem cell concept or model could be a nice explanation for this heterogeneity. For those of you who are not familiar with cancer stem cells, they're a subpopulation of cells that sit at the apex of the tumor hierarchy. They can undergo both asymmetric or symmetric division, always giving rise to cancer stem cells, but more generally undergoing asymmetric division, giving rise to all the other tumor cells we find in the tumor, the bulk tumor cells. So we have here at the apex a lot of pluripotency, high chemoresistance, metastatic nature, while the more differentiated cells are obviously more differentiated and a lot more heterogeneity here. But we believe the cancer stem cell is a very important cell that we need to study. And 31 years later, so take into consideration, we discovered cancer stem cells in 1994, John Dick and colleagues. Where are we today? They've been identified in just about all solid tumors. However, many questions still remain. One, for example, where did these cells come from? What is their exact roles in the tumor? And I think from a clinical perspective, and what we're more interested in, if we eliminate them, can we cure cancer? Now, in order to study cancer stem cells, we have to be able to identify them. So there have been a number of markers that have been published that have allowed us to understand these cells better, isolate them, follow them in tumors, create mouse models, etc. The classical markers that have been used are EpCAM, for example, CD133, CXCR4, ALDH1, and ABCG2. And in the laboratory, we're constantly looking for new cancer stem cell markers that allow us to understand not just the cancer stem cell, but most probably the subpopulations of cancer stem cells that exist within the tumor. CD47 we identified as a marker that allows the cancer stem cells to evade macrophage phagocytosis. P2X7R, FPR2, anthrax toxin receptor, and we're always looking for new markers that can help us understand this heterogeneity that also exists within the cancer stem cell population. And one marker that we published last year is PGLERP1, which can be presented on the cell surface of the cancer stem cell, which can also be secreted. So it also has both extracellular effects on neighboring cells. So this is work that was carried out in the laboratory by a former PhD student, Juan Carlos Lopez Gil. What we saw in the cancer stem cell enriched populations is a number of genes that are upregulated. Here we were able to identify PGLERP1 for the very first time, amongst other genes that have also been associated with cancer stem cells and other types of tumors. And one of the many experiments that Juan Carlos carried out within the laboratory, which is what we published last year, is, for example, if we look at the tumorigenic capacity of KPC cells, so these are pancreatic ductal adenocarcinoma cells derived from a mouse model, some of the mouse models that Meritxell explained earlier. What we see is that wild-type cells and cells where we overexpress PGLERP1, there's really no difference in tumor initiation or tumor take. However, when we knock out or eliminate or genetically edit PGLERP1, these cells no longer have tumorigenic capacity in immunocompetent mice. However, if we take the limiting dilution, here 1,000 cells, and we inject these in immunocompromised mice, we see now that the PGLERP knockout cells are able to form tumors, really indicating to us and all the other experiments to validate this as PGLERP is necessary for cancer stem cell immune evasion. So cancer stem cells express a number of markers. Some have specific functions, some have immune evasion functions. As I mentioned earlier, CD47 is here to allow cancer stem cells to evade macrophage phagocytosis. We believe that PGLERP expressed on the cell surface or even perhaps secreted allows cancer stem cells to evade T effector cell mediated cytotoxicity. We have also discovered, Juan Carlos has discovered, is that secreted PGLERP and PGLERP signaling within the tumor as a bulk entity also promotes subtype switching. So you can see here in this experiment, here we have control cells that are injected orthotopically into mice. We have a doxycycline-induced system where we can treat the mice with doxycycline, inducing the expression of PGLERP. When PGLERP is overexpressed in the tumor, what we see is a reduction in EpCAM or an increase in vimentin, suggesting a transition or a switch to a more mesenchymal or more basal-like tumor. We also see this as a reduction in GATA6 expression. Here you can see at the transcriptional level in 2 KPC cell lines with the overexpression of PGLERP, we have a decrease in GATA6. We also see that the cells in general have a very different morphology. They transfer from a more epithelial-like state to a more mesenchymal elongated state when we overexpress PGLERP. Also, at the level of RNA-seq analysis, we see that one of the hallmark genes or gene sets that are enriched are EMT, also motility, migration, etc., suggesting that overexpression or PGLERP signaling is promoting a more EMT-like state. This can also be seen, sorry for the blurry image, it can also be seen at the RNA-seq level. We can see here that cells that overexpress PGLERP have a negative correlation with the classical subtype, also have a more positive correlation with an EMT more invasive subtype. So this is all suggesting that in addition to allowing cancer stem cells to evade immune evasion, specifically T cells, we also believe that PGLERP is a mediator or a factor for subtype switching. So these are the many markers we discovered. I'm going to show you a marker that we discovered quite some time ago in 2014, led by Irene Miranda when we were at the CNIO. We named this cancer stem cell biomarker autofluorescence. We've been taking advantage of this biomarker for quite some time. What we discovered is that these are discrete vesicles within the tumor that accumulate a substance that give these cells a green appearance or fluorescence green. You can see here in a confocal microscopy these discrete vesicles. This also allows us to not only see these cells by immunofluorescence, but also by flow cytometry, which allows us to track these cells quite nicely. Autofluorescence, what we discovered is that these autofluorescent vesicles are covered in ABCG2. ABCG2 is an ATP-dependent pump. These pumps are pumping something inside of these vesicles, which we later identified to be nothing more than riboflavin, vitamin B2, when at high enough concentrations and excited with a blue light, give a fluorescence color, which is a very nice marker because it allows us then to detect these cancer stem cells within tumors, digested tumors, without the need for antibodies. And you can see that the development of the autofluorescent vesicle is a quite dynamic process. You can see here in live microscopy that the autofluorescent vesicle appears. This can stay within the cell for quite some time, can disappear, and other cells can take on this phenotype. Now, was this something just specific for pancreatic cancer? So we extended our studies to analyze 145 tumors over the course of 5 years, from 2015 to 2019, in collaboration with the Hospital Universitario Infanta Sofia. And what we're able to find is that indeed we can find autofluorescent cancer stem cells in resected digested tumors of many different types of tumor entities, PNETs, PDAC, of course, and colorectal cancer. You can see here that there are some patients that have very high levels of autofluorescent positive cells within their digested tumors, for example, in rectum and also in colorectal cancer. Now here was quite interesting because here at the end of the study we had 75 patients with colorectal cancer in this group, and enough years had passed to see whether or not there was a difference in relapse. 5 years, typically 25% of colorectal patients will relapse. So we were able to divide these patients into two groups, one group that had very high levels of autofluorescent positive cells, another group that had very low levels of autofluorescent cells. And we're going to look at a number of different features, but I think what's really quite interesting here is the probability of recurrence. What we're able to see is that those patients that had high levels of autofluorescent cells at the time of surgery and also positive lymph nodes had a higher probability of recurrence compared to those that had no positive lymph nodes or low autofluorescence, and even those that had just positive lymph nodes, which is the typical characteristics that surgeons will use to predict whether or not a patient has a higher probability of relapse. So combining these two together gives us a much higher possibility of identifying these patients. With this information in hand, we've now trying to establish a multi-center study to look at the presence of autofluorescent cells also in resected PDAC patients. These are patients that can opt for surgical resection to see whether or not that can be a predictor of relapse after surgery. So again, coming back to the autofluorescent vesicles, it's a quite interesting vesicle. So here this vesicle is accumulating riboflavin because obviously riboflavin is a ligand for ABCG2 for these ATP pumps. So we asked whether or not we could take advantage of the fact that they're concentrating riboflavin within the vesicles. Could we hijack this property? Maybe insert something inhibitory, such as a Trojan horse effect into these vesicles to specifically target the cancer stem cells. And this is a collaboration with Maria de la Fuente in Santiago and Adrian Palencia, which is a former postdoc in the laboratory. What we decided to do is instead of using riboflavin, what would happen if we use competitive riboflavin analogs? These are competitive inhibitors. They compete with riboflavin. They can also go into the vesicles or into the cells. So we wanted to see whether or not these different competitive or riboflavin analogs that exist could have a potential to affect the cancer stem cell population. So we primarily focused on lumicrome. And what we can see here is that if you treat the cells with just lumicrome by itself, we have a reduction in the autofluorescent cells, no cytotoxicity, but it's not a great effect. However, when we combine lumicrome or encapsulate lumicrome into nanoparticles, we have much better internalization, no cytotoxicity, and a reduction in the cancer stem cell population. We translated this to in vivo experiments, many in vivo experiments that have been conducted with these nanoparticles. These are orthotopic implantations here of human PDX-derived cells into the pancreas of a mouse. These are immunocompromised mice. These are human cells. The tumor grows. And then after 3 weeks of tumor growth, we start treating the mice with the nanoparticles that contain lumicrome plus and minus gemcitabine. And what we're able to see is the Lumichrome nanoparticle group all had an effect that we're able to reduce tumor size and tumor burden almost by half just with these nanoparticles. Gemcitabine obviously had an effect, but the combination of both gemcitabine and the nanoparticles with Lumichrome had a much more significant effect. So we believe definitely that we can hijack the capacity of cancer stem cells to accumulate riboflavin, either using riboflavin analogs, perhaps using riboflavin attached to a compound, et cetera. But we believe that this is an exploitable characteristic of cancer stem cells that we could potentially take advantage of. So at the omics level, again, focusing on cancer stem cells, over many years we've been trying to understand cancer stem cells at the RNA-seq level, single-cell RNA-seq level, et cetera. We performed a number of analyses to separate non-cancer stem cells from cancer stem cells, comparing them to embryonic stem cells. We've done a lot of methylation profiling, microRNA profiling as well, and again, RNA-seq profiling. And all of this information over the years has been able to allow us to generate a list of putative targets defining what would be a cancer stem cell enriched tumor compared to a non-cancer stem cell enriched tumor. And so we have what we call our cancer stem cell signature versus our non-cancer stem cell signature. We define 112 genes that overexpress in cancer stem cells. 101 genes are underexpressed, or you consider this, these are the 101 genes that define a non-cancer stem cell enriched tumor, and these are the 112 genes that define a cancer stem cell enriched tumor. And if we look at all the publicly available databases to see whether or not we can actually classify patients or tumors as a cancer stem cell enriched tumor or a non-cancer stem cell enriched tumor, we can do this quite nicely. These are just 5 different publicly available databases: Bailey, Fias, TCGA, Junkie, and Pacorread's metadata set. And you can see what we see consistently, and I think a lot of people see this, even when you classify patients as either classical or basal, what we see is the cancer stem cell enriched tumors have a different metabolism compared to the cancer stem cell the non-cancer stem cell enriched tumors. These tumors are very dependent on oxidative phosphorylation, fatty acid metabolism, while the non-cancer stem cell signature tumors are more dependent on glycolysis. So this allowed us to think, then could these differences, if we're able to classify tumors based on whether or not they're cancer stem cell enriched or not, can we use this as a target, or can we apply a specific type of therapy? Drug therapy, and also based on these data, very focused on metabolism. So for a number of years, we've shown already that cancer stem cells use oxidative phosphorylation or mitochondrial respiration to meet their energy requirements compared to non-cancer stem cells. We published a number of articles to show this. And the take-home message is that cancer stem cells are very different than other tumor cells within the tumor at the metabolic level. You can see here that the mitochondria of cancer stem cells are quite functional, very nice defined cristae, very dense images, while we look at the mitochondria from non-cancer stem cells, which are quite swollen, many of them undergoing mitophagy. These are non-functional mitochondria. These cells really depend on glycolysis, while the cancer stem cell have very functional mitochondria and depend more on OXPHOS to meet their energy requirements. So this led us to team up with José Luis Mascarenhas in Santiago de Compostela, who is an organic chemist who develops a lot of metal-based compounds that target DNA. He had this specific compound, IGN-111, that we started to use because of its potential ability to bind to the mitochondrial DNA. So we thought that this could be a very specific or nice metabolic inhibitor, something that had not been tested in pancreatic cancer to date. So Sonia Laboratory was able to show that indeed this IgM111 compound was able to target cancer stem cells and reduce oxidative phosphorylation quite significantly in these cells. Sorry for the blurry image again. Again, the IgM111 will be able to show as it binds to the D loop of the mitochondrial DNA. When it binds to the D loop, it covalently binds to this region, inhibits transcription from the polymerase, so none of the 13 protein coding genes from the mitochondrial DNA are transcribed. And you can see this here on the right. These are just 3 of the 13 mitochondrial encoded genes, 2 different PDX cell culture models. And you can see the very nice dose-dependent downregulation. While if we look for genes that also play a role in electron transport chain and OXPHOS encoded by the nucleus, we do not see this downregulation. So it really seems to be that IgM111 specifically targets the mitochondrial DNA, which translates into a reduction in OXPHOS. We also see a very nice specific accumulation. So if we use our markers, for example, CD133, we see that the IgM111, so this is IgM111 with a fluorescent probe, we find accumulation in the CD133 population, the autofluorescent population, and the CD133 autofluorescent positive double population. So also seems to be nicely accumulating in the cancer stem cell population. In vivo, what Laura was able to show with 3 different models is the ability of this compound to be either very cytostatic or to reduce tumor formation. So what we're able to see here in hindsight is that this is a tumor that's very cancer stem cell enriched, responds very well to IgM111. This would be more of a hybrid tumor, and this would be more of a non-cancer stem cell enriched tumor, which still is a response, but not as effective as in the cancer stem cell enriched tumors. The compound definitely gets to the tumors as ICP analysis showing accumulation of the compound within the tumor. We also see a reduction in the mitochondrial encoded genes within the tumors that are extracted from the mice and treated with the compound. And we also see a downregulation in the cancer stem cell population when we inhibit OXPHOS with this compound. You can see here that the EpCAM positive population is reduced, the EpCAM positive CD133 population is reduced, and this is a triple combination markers that we use to really look for metastatic cancer stem cells and also sees a very nice elimination or downregulation of these cells. So this is IGN-111 in a snapshot. We have the cancer stem cells that sit at the apex of tumor hierarchy, give rise to non-cancer stem cells that have limited cell division or no tumor formation. The cancer stem cell, which has a very high propensity for metastasis, unlimited cell growth, and a very high chemoresistance, which after chemotherapy is really the cause for relapse and the reappearance of the tumor. These cells are very glycolytic, while these cells here are very OXPHOS dependent because these cancer stem cells have a functional mitochondria that we can potentially target, for example, here with IGN-111, which again binds to the D loop, inhibits OXPHOS, stemness, tumorigenicity, which translates into nice preclinical data showing the efficacy of this compound. Fortunately for us, which we're very grateful, is that with this information or with these data, we were able to solicit an EIC transition grant, which we were granted, which is called MEDICS, the goal of this grant is to take this to first in human, so right up until a possible phase 1 clinical trial with IGN-111. So with this, I think I'm ahead of time, which is good because then we have Justo coming next, so I'll give him some of my remaining minutes. So in the laboratory, we're looking at all of these different angles to try to understand pancreatic cancer from a cancer stem cell angle. I haven't talked to you today about precision medicine, but this is also something we're trying to do in the laboratory to more define each patient and see how we can potentially offer different types of therapies. So with this, I would like to end. Obviously, I need to thank my group, which is right here, both present and past members and new members that are joining tomorrow. And of course, our collaborators, which are very important, both at La UAM, María de la Fuente, Alfredo Carato, Julie, Laura, and EDCs, Hospital Infanta Sofía, José Luis at the University of Santiago de Compostela, Susana Garcia at the CNIO, and of course, our funding agencies, because without them, we would not be able to perform, I would not be here today showing you these data. So with that, I would like to thank you all for your attention. I'll be more than happy to take any questions.
So you heard Bruno, questions. Do you see tumor resistance to your compound?
It depends on the tumor.
On the PDX?
It depends on the type of PDX. So we would probably have more chemo resistance or more resistance to the compound if it was a non-cancer stem cell enriched tumor. So you definitely have tumors that are more, obviously there's heterogeneity you don't want to classify. We know that tumors aren't 100% basal or 100% classical. There is heterogeneity, there's mixing of these cell types within the tumors, but we find that there are differences and we can correlate those differences to what we call our cancer stem cell signature.
Oh, it looks pretty good.
Yeah. Great presentation.
Thank you.
In pancreatic cancer patients, they usually have. Hello? Can you hear me? Yes. So great presentation.
Thank you.
Our pancreatic cancer patients usually have high burden of disease, and the cancer stem cell population is a relatively small fraction of those, right? So any treatment that it's just targeting those in patients who have short life expectancy is not likely to give us a meaningful response to really extend life expectancy. Have you done any studies with this molecule combining with chemotherapy in the lab?
We have. I haven't shown you that today. We've done some limited studies. This is also part of the MEDICS project to extend upon that, to combine with gemcitabine Abraxane, to combine with FOLFIRINOX, and to combine with some experimental drugs. What we've done already, we do see synergy. So in that essence, by attacking both the cancer stem cell population and also the non-cancer stem cell population, most likely with the chemotherapy, there we do have a synergistic effect. And it's not just for pancreatic cancer. We've also seen very nice effects in colorectal cancer, osteosarcomas, also in breast cancer. So it seems to be something that could be applicable to many types of tumor entities.
Okay,
Can you hear me? Okay.
Yes.
So this is following up on Ignacio's question. So we know, at least in the context of KRAS inhibition, that you have a switch from glycolytic dependency to more like OXPHOS and fatty acid oxidation dependency. So, I mean, the obvious question here would be, why not combining the KRAS inhibitors with this? Either but to exacerbate, that dependency on the OXPHOS in the cancer stem cells, but also on the remaining cells, to make them transition to this dependency that the cancer stem cells already have. So.
No, a great suggestion, something that we also have in mind. There are a number of ways to sort of target this. If we have a tumor that's already, if we have an inhibitor that targets cancer stem cells or OXPHOS in general, is there something we can do to push the tumor to a more OXPHOS state? Therefore, the therapy would be more effective. Or some type of subtype switching, et cetera. So that would be a good approximation. If you're treating with the KRAS inhibitors and promoting the appearance of more OXPHOS-dependent cells, then the therapy could, in theory, combine or be synergistic with that therapy.
Okay, one more question. If not, we go ahead with the next talk. Thank you very much.
Has a few more minutes. Thank you, Mariano.
Okay, next speaker is Dr. Justo Castaño from the University of Cordoba, down in Andalusia, right? Al-Andalus, that we say now it's become fashionable, right? So the title of his talk is Dysregulated Splicing, a new key player in pancreatic cancer.
Thank you very much, Mariano. Thank you very much, Carmen, and the Fundación Ramón Areces. It's a pleasure and an honor to be here to talk about our favorite topic, which is splicing. And today I will try to convince you that splicing is important in cancer. This is the outline, and let's restart this again. I will talk about alternative splicing as a new hallmark of cancer, the machinery, spliceosome, how it is dysregulated in cancer, how it is dysregulated in pancreatic cancer, and how it provides novel opportunities, even for immuno-oncotherapy. And then some conceptual challenges that we have in studying splicing. I don't need to introduce to this audience the importance of finding new avenues to prevent, to diagnose, and to treat resistant pancreatic cancer. And we need to change, because we have been doing the same thing forever, and it has not been so helpful. We know a lot about the underpinning, the molecular landscapes, the multi-omic landscape, the single-cell omics of cancer, of pancreatic cancer, but we still have metastasis, we still have chemoresistance, and we still have not improved the survival of patients. Most of the research in cancer has been driven in the last 25 years through this very prescient conceptual development of the hallmarks of cancer. We came from conceptual to translational to integrative and very comprehensive lately. Although I missed splicing here, although RNA is there. Why do I talk about splicing? Because splicing is pervading all these hallmarks of cancer, every one of them, every single one of them, and it is still not considered a hallmark of cancer. And why is that? Because RNA is the Cinderella in the flow of information in the classic dogma. How much we know about DNA? Oh, a lot. About proteins? Wow, a lot. What about DNA? Have you heard about long non-coding? No. Messenger RNA, ribosomic RNA, transfer RNA. Long non-coding is recent, circular is recent, PIRNAS is recent, splicing is recent. We have invented the word spliceosomic because it was not before here. This is a constellation of a new layer of control and regulation that has not been explored, from transcription to translation. And there are a lot of mechanisms and machineries, and we have focused in splicing. Why? Because splicing is a central, fundamental mechanism regulating gene expression in eukaryotes. It provides a means to make multiple types of mRNAs or other types of RNAs from a single gene, and this is very versatile. And what is the machinery that does that? This is it, the spliceosome. Now we know how it is. And how it is? Well, it's quite similar to a ribosome, except that the ribosome, small particle, large particle, that the two subunits bind together, and they're very stable. And the spliceosome is not. It is a cycle of activity that goes from the start to the end, changing plastic in a plastic and dynamic manner. And it's not very formal. It's more probabilistic. It is not very precise. It has more noise. And in doing so, it is utilizing more than 300 players, which we call splicing factor, which are promiscuous because they play in another fields, not just splicing. And this is quite important because this dynamic cycle will provide the means to the cell to be this way or this way, in a different manner. Now, is this important? Well, it is defined as a fundamental mechanism like transcription or translation, except that it has not been so well studied. And because it is so fundamental, when it goes wrong, it is quite important. This regulation of splicing is linked to diseases, and particularly to cancer. Why? Well, imagine by changing splicing, you can make a tumor suppressor to be a tumor inducer, to make a molecule from pro-apoptotic to anti-apoptotic, to be an adult isoform or an embryonic or tumoral isoform. It can be angiogenic or anti-angiogenic from the same gene. It can be tumor sensitive or tumor resistant. It can be anti or pro-apoptotic. So splicing is a key element in controlling what a gene does and what a gene means. And this is why it has become a flourishing, a blossoming field. Splicing in cancer is now regarded as a novel hot topic in cancer, and many reviews have significantly improved our knowledge about the relation of this co-transcriptional mechanism, because splicing goes exactly at the same very moment of RNA polymerase transcribing the mRNA. Is there with other machineries. So, indeed, splicing is, and its alteration, a novel hallmark of cancer, pervading all other hallmarks of cancer. And how can it contribute? Well, in different manners. For example, it can work on the initiation of the tumor, in the progression, or in the response to therapy. And who plays that role? Well, the splicing machinery itself. It can be mutated, as it has been shown by a number of oncohematologists that are always advancing the field of research, like SF3B1 mutations in leukemia, or altered expression. You don't need to have mutations. You just need to have a wrong level of splicing factor. Or you can have subsequently and consequently changes in the pattern of splicing factors. And this will change the fate of the cells. So we ask ourselves, because we were studying a somatostatin receptor was weird, it was a 5 transmembrane receptor instead of 7. And we wonder, is the splicing machinery wrong? We made a very candid approach. We use an array to develop an expression measurement of a number of genes in the core of the spliceosome and splicing factors in a number of tumors. We started in pituitary because it was our subject at that time. Then we went to prostate, to pancreatic neuroendocrine tumor, pancreatic cancer, and see what? The machinery was always wrong compared with the normal tissue, always altered, always significantly altered. We teamed with an artificial intelligence expert, with Sebastián Ventura, and he helped us to select those genes that were most likely responsible for the changes that we were observing. And we have done this in a number of tumors. And there are several teams now working on this in particular types of cancer, like liver, like prostate, like brain cancer. But we continued with pancreatic adenocarcinoma and pancreatic neuroendocrine tumors. And is the machinery wrong in pancreatic cancer? Is this actionable? Is targetable? Well, believe me, it was one of the pioneering fields, pancreatic cancer. Larry Miller was developing 15 years ago the concept that the spliceoforms, the machinery, was altered in pancreatic cancer, and then the blossoming. We have witnessed in the last years a number of significant studies demonstrating that the machinery of splicing is altered in pancreatic cancer. This was our modest, humble contribution. We followed the path that I showed you just a minute ago to discover that 1/3 of the machinery was completely dysregulated compared with normal pancreas in patients. And particularly, we selected 2 of them to study in more detail. And what we observed is that the repression of these 2 factors was significantly linked to clinical parameters. It was possible to reverse their oncogenic effects by enhancing their expression in a number of models. It was linked to molecular parameters that are linked to the oncogenic properties of pancreatic cancer. And this is just an example. We made a more profound study with SF3B1, both because it is well known, but because it is also actionable, it is targetable. And what we observed, this is a work made by Emi and Ricardo, we observed SF3B1 is one of the core components of the machinery of splicing. It is enhanced, it is overexpressed. It is mutated in a low percentage, just 2% of patients, 1 point something. But it was overexpressed in the tumor. It was related to the overexpression was related not just to our samples, but in other data sets, public data sets. It was related to the disease stage, lymph node, metastasis, so a number of clinical translational significant features. We ask, well, this is dysregulated. Is it related to splicing? Yes, indeed. Just the level of expression, I'm not talking about mutations in SF3B1, is related to changes, significant changes in the profile of the events that we see with splicing. So the genes are being completely changed in how they are processed because of the level of SF3B1. And in this large panel, you will see that the level of SF3B1 is not so much linked to mutations in key genes, but to the level of expression of these same key genes. So we have abandoned the level of changing the genes by mutating them to changing the genes by modifying the level of expression or the subtype of isoforms that we change. Now, can this be targeted? Yes. SF3B1 is the leading compound, the leading component that was targeted by a compound, the pseudomycin, and particularly pladienolide. And pladienolides enter SF3B1 and modifies splicing. It does not completely eliminate the splicing, it modifies the splicing. And what we observe using let's say, normal model pancreatic cells and different aggressiveness of levels of pancreatic cancer cell lines is that silencing SF3B1 was deleterious, was very bad for the cells, including the normal cells. If you use pladienolide, what you see is that the normal cells are not so much affected, but cancer cells, the most aggressive cancer cells, are targeted, and you can kill them. You can decrease proliferation. You can mix this with a chemotherapeutic, and you will enhance the ability of pladienolide to kill the cells. So we have a targetable component of this splicing machinery, and we demonstrated also that another set of oncogenic features, like tumor sphere formation, like clonogenic, like apoptosis, were also targetable using this same compound. And we observed that this was related to the type of signaling pathways that were activated or inactivated, as well as to the genes that were modified. We did not do a complete set of analysis of the modifications in splicing, but significant genes like BCL isoforms, KRAS isoforms, or TP53 isoforms were modified because of the treatment with pladienolide. So it was through a splicing alteration that it was working. And we were fortunate enough to collaborate with Bruno to see whether this was also related to the cancer stem cell population. And in fact, it was present, SF3B1. It was altered, and it was modifiable in cancer stem cells. For example, SF3B1 was able to modify the phenotype, let's say, of cancer stem cells. And it was able to be related to the self-renewal and tumor suppressor genes that are expressed in these populations. Moreover, the treatment was able to reduce stemness of the cancer stem cells. We were making them less stem cells. And even the apoptosis was induced in these very highly chemoresistant cells. This was also shown in vivo in using a xenograft model with MIA-PACA cells, and it was also shown both with MIA-PACA and with cancer stem cells in a zebrafish model. So targeting splicing and targeting the machinery is a vulnerability of pancreatic cancer, which is highly dysregulated in pancreatic cancer. But this contribution was just the tip of the iceberg. In the meantime, we have seen RB-FOX being published in Nature because it is related to metastasis. We have seen RB-FOX related to cancer progression. We have seen Quickie related to basal-like splicing signatures. So these groups are establishing, they are supporting that splicing is the one that determines if you are a basal-like signature PDAC. That's major words, and it's not ours. So there is a lot of activity going on in the field, and you can follow this, for example, in this very nice review where all the properties that I have been going through is very nicely summarized on the importance of splicing dysregulation with particular molecules for EMT, proliferation, metabolism, the different studies that have demonstrated the role of SF3, SRS, or SF1, RB-FOX2, Quickie, and the field continues advancing. SF3B1, our study, which is summarized here, is one of them. It is related, highly related to hypoxia, which is very strongly linked to splicing dysregulation too, and mutations are considered a potential targetable cause of pancreatic cancer. Now, pancreatic cancer can be splicing in pancreatic cancer in other cancers can be targeted not just with pladienolide. there are other splicing inhibitors, but there are also antisense oligonucleotides. There are also isoform-specific targeting, SIRNAs, and there are other type of approaches. So it is a new vulnerability. It's a new tool that can be used. But, scientists, how we are. We see something new, and we all turn our eyes. I was studying immuno-oncotherapy, but then I saw splicing-derived neoantigens, and wow, that's an opportunity. In fact, it is. There is a blossoming of studies related to neoantigens as targets for immuno-oncotherapy. Now, this is being strongly considered as a novel tool for cancers that are regarded as cold for immunotherapy, because you can always find some precise neoantigens. And the basis is that you change the type of peptides that are formed, either because you provide a new, let's say, embryonic type of neoantigen, or because the splicing is dysregulated, is aberrant, and then you have a new peptide. And this new peptide can be a new target. This is being strongly developed now from an industry point of view, and there are biocomputational findings, experimental findings to demonstrate that neoantigens are a new source of tools for immunotherapy. And we have in our team the good news that Alejandro is leading this line of research, and he just got a FIS grant to continue the grant that we received from AESPAC and the Asociación Española del Cáncer de Páncreas, and he will continue with this study, which is under development now, providing the tools to change splicing, because when you modulate the splicing, the number and type of neoantigens can be enhanced, and you will have more targets for immunotherapy, because modulating splicing is not good just for neoantigen and vaccines. It's good for many other things, and this is being developed and understood now. And believe me, even though some teams and some governments are saying that mRNA vaccines will not be developed, they are here to stay and succeed. And these are three wonderful examples: 2017, 2023, 2025 of vaccines derived from neoantigens and mRNA. And let me finish in 2 minutes talking about a conceptual challenge. If we want to understand why splicing is important in pancreatic cancer and other cancers, we need to develop novel conceptual tools. And we have called this spliceosomics. Spliceosomics means that I measure the machinery that makes splicing, the spliceosome, all these 300 and something splicing factors, and we measure the variants. And then we have a picture of how splicing is in a normal cell, in a cancer cell, in different stages, in response to this and that and that. So this is a novel tool. Using this, Ricardo Blázquez and Encinas Rey, who is now in the Botón Champalimaud with Vincenzo Corbo in Lisbon, made a study on pancreatic neuroendocrine tumors. What he found is that there are 3 subtypes. They are not G1, G2, G3. They are different, but they linked with the known, very tightly, with the known major mutations. MEN1 is highly related to splicing dysregulation, and we're studying that. Moreover, he found that these 3 types of neuroendocrine tumors from the pancreas were completely different in terms of the targets of the current treatments, somatostatin receptors, mTOR inhibitors, etc. Moreover, the microenvironment was different in these 3, and they were only discovered through their splicing machinery. But most importantly, in my mind, is what we found. This might seem obvious. Splicing machinery drives splicing events. Oh, well, of course. Yes, well, it was not demonstrated before. If you characterize these tumors using splicing events, not the machinery, the splicing events, the 3 types that you obtain are the same, almost 3 types with the machinery. So there is a link. And why is this important? Because these 3 types determine the fate, the biological fate of the cells, how they behave, what type of isoforms they generate, what type of mechanism is in there. So there is a lot of work to do. But at least we have now the Google Maps to work with the splicing. Juan Valcárcel and Gosia Rogalska published in Science this year the cartography of a splicing machinery in human. And we now can guess what each one of the molecules that we find that is dysregulated is doing and why it can be important. So I hope I have convinced you that the splicing, including the spliceosome components and splicing factor, is dysregulated in cancer and is an informative tool, that the splicing machinery is drastically dysregulated in pancreatic cancer, and for example, SF3B1 is an actionable marker, and that splicing is an emerging mechanism, a promising tool in oncology, providing, for example, neoantigens, and that we need to look at spliceosome. But don't take my word for granted. Go for the giants, the brilliant people. These 3 Nobel Prize couples related to the catalytic ability of RNA splicing or microRNA last year. What he said is, in the past 40 years of research have taught me anything, it is never underestimate this little molecule. The age of RNA is just getting started. So my take-home message is, keep an eye on splicing when you do your studies. Meritxell, Bruno, anyone, look at the splicing. So thank you very much to all the team, to all the collaborators and the funding bodies, and thank you very much to all of you.
This is what I call a humbling presentation. When we thought we knew everything, you come with the splicing. Okay, so questions for Justo, Dr. Castaño? Nothing.
That was amazing. I love this presentation. You were talking about RNA-seq, and it's incredible because just last week we had the first paper showing us clearly that patients with the basal phenotype, PDAC patients with basal phenotype, respond poorly to certain chemotherapy, FOLFIRINOX, while they seem to do better with nab-paclitaxel and gemcitabine. We cannot use this in the clinic yet, so people is looking at other markers, like I heard GATA6 before, right? My question to you, I saw on your slide PRMT5, and this is an actionable target. We know around 20% of patients with PDAC are going to have MTAP loss, and we have a number of phase 1 trials being developed, including combinations with RAS inhibitors. Can you elaborate to educate someone like me who didn't know this connection with the spliceosome on the PRMT5 and the spliceosome? Can you educate me a little bit? I know nothing about it, but I just glanced on your slide.
Okay, this particular molecule, thank you very much for the question. This particular molecule, I don't think has been fully investigated in terms of the splicing isoforms and why is there, but it is linked. What I can say is that most of the results that we are obtaining until recently derived from RNA-seq studies did not pay attention to splicing isoform. You just had one gene, KRAS, but you didn't know if it was A or B, just because it was not focused there. Now we have much more in-depth and biologically and technically deeper analysis, and we can now work with isoforms. The isoforms may provide critical information of why one patient responds or not to a chemotherapy. Moreover, the new, I would say, application of long-read sequencing to study changes in transcriptional expression of genes will provide a more detailed picture of the variants that are altered in the different responses. So I think that every time that you get a gene that is important for a response, that is actionable, it is good to look, just doing a little search on whether there are isoforms, whether there are variants, and why this can be important in your research.
Okay, one last question. One or two quick ones.
Okay, so related to the splicing events, what do you think is regulating them? Are they affected by the different, genomic alterations? How major drivers such as KRAS are contributing, or major tumor suppressors are contributing to this? Is this just a consequence of the tumor evolution and just the oncogenic, and then the tumor phenotype, but not the oncogenic inducer? So what do you think is the major player there?
That's an excellent question. Thank you, Silvia. The hierarchical regulation of the splicing machinery has not been defined yet, but the map that I showed you in Science by Juan Valcárcel is a very nice proxy of it. So we now know what genes are regulated by which transcription factors. And this is a critical information to see how splicing is altered and evolves. I can for example, suggest that cancer heterogeneity has a critical component in splicing alterations. Who drives that? Is first the chicken, the egg? Well, one of the initial moments for one cell to decide what type of tissue you have to do is not just one gene, is the isoform of this gene. So this is going to change in a very mild manner with nuances. It's not that you are disrupted completely, it's that you are changing this little tail. And this can be known now, not 15 years ago, where the techniques did not allow. Now, the transcription factors that regulate the splicing machinery have not been completely deciphered, but I can anticipate that in 10 years from now, we will have a very precise map of who drives splicing as a whole, probably with machine learning and artificial intelligence.
Good afternoon, everyone. Could we start to take our seats and start this next session on preclinical models? Thanks. Thank you. So good afternoon and welcome to this next session. I'm Julie Earle. I'm a basic scientist from the Ramon y Cajal Institute of Research here in Madrid. So in this session we have three great speakers who will share their insights and their research using preclinical models in our fight against pancreatic cancer, from prevention to overcoming treatment resistance. Each presentation will be 20 minutes and we have 5 minutes for questions. So let's begin with our first speaker, who really needs no introduction. It's Paco Real. He's a medical oncologist from Memorial Hospital in New York. He is currently a group leader at the CNIO, using these preclinical models to look at pancreatic tumorigenesis and to determine how the tumor develops. And today he will talk about how we can leverage these models for precision prevention of pancreatic cancer. So thank you, Paco. Sorry, I've passed your slide there.
Thank you, Mariano and Carmen, for the invitation. Thanks to the Fundación for the organization of this great meeting. We're here because of a war, the war against pancreatic cancer. And this is, unlike other wars, this is a fair war, it's a must war, and it's a formidable war. Let me see, where do I have the pointer? Or do I have to? Okay. No pointer? Yes? Where is the pointer? Oh, here. Yeah. Okay. So, this is a war. It's so formidable that it requires land, sea, and air. And land, sea, and air means tertiary prevention, treating the cancer, secondary prevention, detecting the cancer early, and primary prevention, preventing the cancer. Primary prevention, preventing the cancer. And these are two mottos or three mottos that have guided the research in our lab for the last 10 or 15 years. The first one is, in pancreatic cancer, early is too late. We said this about 25 years ago. A 2-centimeter pancreatic tumor is most likely already a metastatic tumor. This is unlike any other tumor. The second important concept that has guided our work and that has led us to focus on prevention is that it's obvious that we need better drugs, but we need more than better drugs. 30% of patients with pancreatic cancer have cachexia at presentation, and therefore this limits dramatically the amount of drugs they can take. So we need to tackle this disease in a comprehensive manner, and we think that whether we can prevent cancer, pancreatic cancer, and whether we can apply precision prevention is another of the weapons that we can use in this war against pancreatic cancer. Well, if we want to prevent cancer, we need to know what is it that we have to avoid. And these are the causes of pancreatic cancer, what we know about the causes of pancreatic cancer. And I want to emphasize that many of these causes, or these factors that are involved in the risk of pancreatic cancer, confer a relatively small increase in risk, no more than 2.5-fold. We have tobacco, we have alcohol, we have obesity and diabetes, metabolic syndrome. We have chronic pancreatitis, and we have family history. Most of these risk factors are associated with an inflammatory-related processes. Therefore, we have decided in the lab over the last decade that we're going to try to understand these very early steps of pancreatic cancer development, which are the ones where we would be able to impact in terms of prevention. And our strategy, because we cannot study these very early steps in humans, because the pancreas is very difficult to access and we don't know who's going to get pancreatic cancer, is that we make observations in humans, we go to mice, and then from mice we try to bring back those observations, those discoveries we've had in mice, to see whether they are relevant to humans. This has been the approach we've taken. And I must say that not so many people have been interested in taking this approach. So the first risk factors against pancreatic cancer are the genes we inherit from our parents. And therefore, we have dealt with some of the GWAS hits that have been identified by the consortia and in collaboration with Nuria Malatz. And I will tell you today about two stories that are focused on two genes where genetic variation in these genes has been shown to be associated with an increased risk of pancreatic cancer. And we want to know how these genes contribute to pancreatic cancer and whether we can do something about it. The first one is NR5A2, the second one is CTRB2. And I just want to emphasize again that these genes, these hits that have been identified through the GWAS, are involved in processes that are very important to understand how the normal pancreas works. Differentiation, cell differentiation, particularly differentiation of acinar cells, Meritxell told you about earlier. ER stress, which is an intrinsic feature of acinar cells because of their high protein synthesis capacity, and inflammation. The first gene I'm going to talk about is NR5A2, or LRH1. It is an orphan nuclear receptor. It is involved in pancreatic homeostasis. It is required for pancreas development and acinar differentiation. It is required for the complete differentiation of acinar cells, and we think that differentiation is the first tumor suppressive mechanism in a tissue. NR5A2 also participates in other functions in the GI tract, cholesterol biosynthesis and ER stress in the liver, and both NR5A2 and another related factor where genetic variation has also been associated with the risk of pancreatic cancer are part of a differentiation network. Not only that, as I said, variants are associated with the risk of pancreatic cancer, but also of pancreatitis. This is unpublished work from us. And we know that these processes are important because genetic variation in other genes that are involved in these processes are also contributing to pancreatic cancer risk. So we hypothesize that these very common genetic variants, which are the ones identified in GWAS, would not be gain of function mutations or variants, that they would probably be partial loss of function variants. And therefore, we decided to study heterozygous mice, NR5A2 heterozygous mice, having only one allele. And as you can see, the pancreas, I really dislike this pointer because it's delayed in time. Anyway, heterozygous mice have a completely normal, histologically normal pancreas. And despite the fact that they have completely histologically normal pancreas, they don't pass what we call the stress test. The stress test in pancreas physiology is like the stress test in cardiology. We were inspired by the stress test in cardiology. If we give cerulein and induce a mild acute pancreatitis to NR5A2 wild-type mice, at 24 hours we have edema, we have inflammation. By day 5, the inflammation is completely resolved. NR5A2 heterozygous mice, which have a completely normal pancreas histologically, they don't pass the stress test because when we administer a low dose of cerulein, they don't recover properly from pancreatitis. Not only that, when we cross these mice to introduce a KRAS mutant allele and we induce pancreatitis, we have increased occurrence of preneoplastic lesions and PDAC. Therefore, this mimics exactly what happens in human. A genetic susceptibility variant, in this case, heterozygosity, a somatic mutation, KRAS, and an environmental or lifestyle factor, alcohol in humans, pancreatitis induced by cerulein in mice. So we asked ourselves, if the pancreas is histologically completely normal, what is it that makes these mice susceptible to pancreatitis and to pancreatic cancer? And for this, we turned to RNA-seq. We performed RNA-seq in basal conditions, and then 8, 24, and 48 hours after administering cerulein. And what you can see here, and I want you to focus on the circles and the triangles, and I want you to focus on the purple and the orange, is that the heterozygous mice, triangles, which are in basal conditions at 0 time, their transcriptome is related or is similar to the transcriptome of normal mice in which a pancreatitis has been induced, and we look at the pancreas at 8 hours, when the full pancreatitis is not in. And we say that this is pre-inflammation. These mice, their pancreas does not contain leukocytes, inflammatory cells, is not inflamed. However, there is something transcriptomically that suggests that this pancreas is not normal. And indeed, when we do gene set enrichment analysis of the transcriptomic analysis of this pancreas at 8 hours, what we can see, in basal conditions, what we can see is increased activity of a number of pathways that are related to immune response, defense response, humoral immune response, inflammatory response. And many of these genes are actually related to chemotaxis. And you can see here several complement components, several chemokines involved in chemotaxis. Therefore, this is what we said, pre-inflammation. And we then looked at NR5A2, which is a transcription factor that regulates acinar differentiation. How could it regulate inflammation? I'm summarizing here a lot of work carried out by Isidoro Cabo and a few other colleagues in the lab. And what you can see here is cheap PCR on 3 groups of genes. Acinar differentiation genes, which in this case are digestive enzymes, all of these in black, AP1 proteins in orange, and inflammatory genes in blue, the ones that I showed you here. And as you can see, in wild-type mice, in basal conditions, NR5A2 is sitting on the promoter of genes involved in acinar differentiation. NR5A2 is doing what it should do, promote acinar differentiation. Yet, in heterozygous mice, which express 50% of the dose of NR5A2, NR5A2 is preferentially found on AP1 and on inflammatory genes. And this mimics exactly what happens when we administer a single dose of cerulein. You can see here this is basal conditions. The first column in this graph is copy-pasted from here. In basal conditions, NR5A2 is sitting on digestive enzymes, acinar differentiation. We give cerulein, and half an hour later, NR5A2 has translocated, so to say, to genes that are involved in the control of inflammation and to AP1. And 24 hours later, it goes back to differentiation genes. So actually, what this shows is that NR5A2 is a transcription factor that at the same time promotes cell differentiation and suppresses inflammation. Is this relevant to humans? Well, what we did is in this transcriptomic comparison of heterozygous versus wild-type mice, we identified 1,000 genes that were differentially expressed, and 900 of those were upregulated. We took those 900 genes and we looked how they were expressed in individuals from GTEx, normal pancreas from organ donors, GTEx, low NR5A2, mimicking the heterozygous, high NR5A2, mimicking wild type. And 68% of the genes that were upregulated in the heterozygous mice in normal pancreas were also upregulated in the normal pancreas of individuals with low NR5A2 levels, strongly suggesting that what we found in mice is relevant to humans. If it's not relevant to humans, we're not interested in it. So the two major messages here is that the pre-inflammatory phenotype is driven by epithelial intrinsic mechanisms. I didn't quite show you that, it's in the paper. And that transcriptional networks involved in cell differentiation also suppress epithelial cell intrinsic inflammatory programs. So how is pre-inflammation established? And we've looked at a number of things. I'm just going to give you a hint of some of the stuff that we have done recently that is not published yet. But we thought that maybe the gut microbiome might be involved in contributing to this pre-inflammatory phenotype in the pancreas. So what we did is we treated mice with antibiotics for 1 week with a cocktail of antibiotics that pretty much depleted the gut flora. We waited, and then we administered a week to 3 weeks, and we administered cerulin and sacrificed. And what you can see here is that when we do that, when we administer water, this is the pancreatitis that we can see in heterozygous mice, very severe. But when we administer antibiotics and deplete the gut microbiota, this phenotype is completely rescued. So I'm telling you that we are rescuing a genetic defect with an intervention, which in this case is antibiotic administration and gut microbiome depletion. When we take these mice that have been treated with antibiotics and we reconstitute with poo from the mates in the same cage, we recover the pancreatitis phenotype. Therefore, this clearly shows that it is the microbiome that is responsible for establishing this pre-inflammatory phenotype in the pancreas. And this can be shown here with more quantitative assays, plasma amylase and histoscores. So, in order to understand how antibiotics might be acting, we again turn to RNA-seq, which is a good discovery tool, we think. And what you can see here is that when we treat mice in basal conditions, no pancreatitis, with antibiotics, and we compare mice that received water versus those that received antibiotics, we see that there is increased activity of pathways to protein synthesis and mitochondrial function. That's precisely the main function of acinar cells, producing acinar genes, acinar digestive enzymes in huge amounts, and decreased activity of pathways related to coagulation, ECM, and inflammation. Therefore, we think that what antibiotics do is they induce a sub-inflammatory state, and we think that the gut microbiome establishes an inflammatory tone systemically. We've only looked at the pancreas, but there's no reason to think that this wouldn't happen in other tissues. And one of the hypotheses we are testing is that this might be mediated through metabolic factors produced by the microbiome. I'm going to summarize much of the work that has been done in the last 2 or 3 or 4 years in the lab, but I'm just going to show you that we've come to the conclusion that, and this is still ongoing work, that tryptophan metabolites play a very important role in this process. This is the way tryptophan is metabolized by the microbiota in the gut, the indole pathway, the kynurenine pathway, and the serotonin pathway. When we looked in mice treated with antibiotics and reconstituted at the serotonin or at the kynurenine pathway, we didn't see differences between control and antibiotics treated mice, but there is a very clear change in several metabolites related to the indole pathway. And what we are trying now is to identify those metabolites, actually show that they are causal, and this is work that is ongoing, and if so, we will try to interfere selectively with those pathways that are involved in the generation of these metabolites. So the working model is that we would have signals coming from the microbiota that could be inflammatory directly or metabolic, that they would have systemic effects, and they would prime the tissue for the response to KRAS or to the response to other damage. The second gene I want to briefly tell you about is a more recent work carried out in relationship with CTRB, chymotrypsinogen B2, which is one of these acinar enzymes, digestive enzymes that is produced massively in large amounts in the pancreas. And genetic variation in CTRB2 has been shown to be a risk factor for developing pancreatic cancer. This is a very interesting locus because it's not only involved in the risk of pancreatic cancer, it's involved in the risk of type 1, type 2 diabetes and chronic pancreatitis. So it's a complex and very genetically rich locus. And Lofia Mondatotir, with whom we have collaborated at the NIH, fine-mapped the risk variants identified through GWAS and found that a deletion of exon 6 that occurs frequently in the population, 17% of normal individuals carry the deletion of this exon 6 in CTRB2, might be the causal variant. This deletion was predicted based on the amino acid sequence to lead to a truncated protein that might cause ER stress due to misfolding. So in collaboration with Sagrario Ortega, we've generated a mouse model that lacks the precise exon 6 that I was telling you that is a human germline variant in the population. We actually showed that the exon 6 is not present at all in CTRB2 by RT-PCR in homozygous mutant mice, and half levels are found in heterozygous mice. We showed that wild-type mice express wild-type CTRB1 of about 36 kilodaltons. Homozygous mutant mice express a truncated protein, as do heterozygous mice as well. And not only that, we show that the mutant protein that is truncated partitions in, when we look at solubility in SDS and Rana-Western blot, partitions not only to the soluble factor, but also to the insoluble fraction, suggesting that this protein misfolds and causes ER stress. Indeed, when we look at the pancreas of these mice, it's completely normal, as you can see histologically. When we look at CTRB1, CTRB1 is expressed in zymogen granules, in normal acinar cells, as it should, and these fine speckles that you can see here. However, in mutant mice, we see these massive aggregates that accumulate in acinar cells, again going in the direction that there is misfolding. We see increased activity or expression, of calreticulin, a classical ER stress. And when we've done electron microscopy with Miguel Lafarga, you can see that there is massive dilation of the endoplasmic reticulum. There is accumulation of intracisternal granules very persistently and pervasively in acinar cells and cytoplasmic inclusions. All of this in the context of a completely normal histologically pancreas when we do H&E. So H&E does not really have a lot of resolution to tell us about what's going on in the pancreas. So we've done RNA-seq again in these mice, in basal conditions, in young mice, when there is no phenotype whatsoever. And what you can see here is that we see a dramatic downregulation of the expression of acinar genes and of a more complete acinar signature derived from single-cell data. You can see here in this heat map that the most important and most abundant digestive enzymes are indeed downregulated in these mice, and we interpret this as a downregulation of the program that involves CTRB itself in order to relieve the ER stress caused by the mutant protein. You can see here the accumulation of the granules that I was aggregates that I was telling you later. And when we administer OP-pyromycin to mice, we see particularly in homozygous mutant mice a dramatic downregulation of the synthesis of total proteins. That would be expected from the fact that the cells, we believe, are trying to protect themselves from the massive ER stress caused by the misfolded protein. As I told you, we don't find much evidence of inflammation histologically in these mice at all. But when we do RNA-seq, we again see an increased activity, very increased and prominent activity of pathways related to inflammation. You can see here in the heat map. And you can see that there's a mild increase in the number of CD45 positive cells. Again, very, very subtle. And very massive accumulation of activity of pathways related to ER stress, to the unfolded protein response, and chaperones, all consistent with the presence of these aggregates. Also confirmed, as you can see here, by Western blot when looking at the expression of CHOP or BIP or IRI1, which are the three branches of the UPR. One of the things that was quite striking for us as well was that there was a significant upregulation of pathways related to DNA damage and to apoptosis and of the p53 pathway, suggesting that these processes might actually forecast what could be related to the risk of developing pancreatic cancer in these individuals. And I want to emphasize here that one of the most upregulated genes that we found in the RNA-seq analysis was AGR2. AGR2 is a chaperone. It was mentioned earlier by Meritxell this morning. And we have gone on to try to map, using spatial transcriptomics, how are these alterations taking place in tissue in a spatial manner. What you can see here is in gray are wild-type mice, in fuchsia are heterozygous mice, in purple are homozygous mutant mice. The three different graphs correspond to 3 months, 6 months, or 18 months. And what you can see here is the activity of the acinar signature. On the left side, you see wild-type mice. On the right side, you see a homozygous mutant mice. And you can see that indeed we confirm, looking at the using Visium HD at the acinar spots, we can see dramatic and persistent downregulation of the acinar signature, which means incomplete differentiation, increased ER stress signatures all along time, and increased activity of a signature that we have identified as the delta-delta signature that's characteristic or specific for the delta-delta mutant mice. But most interestingly, we took a signature derived from expressing OSKM, the 4 Yamanaka reprogramming factors in the pancreas. This was done in collaboration with Manuel Serrano, and the signature was obtained 4 days after inducing OSKM in the pancreas. After inducing OSKM in the pancreas, and you can see that starting already at 3 months and continuing and persisting up to 18 months, there is increased activity of a reprogramming signature in acinar cells in the pancreas, suggesting increased plasticity, and we believe that this might set a biological context of increased transcriptomic entropy that might sensitize cells to the effects of other damage, including inflammatory damage or KRAS. Well, as I told you, we're interested in it if it's relevant for humans. So what we did is we went with Nuria's group to GTEx, and we identified in GTEx those individuals who were homozygous or heterozygous for the exon 6 deletion mutation, as well as individuals who were wild type. And when we compare the individuals carrying the variant versus those who were wild type, we can see that, again, in those carrying the variants, we see a reduced activity of the acinar program, as we saw in the mice, and we see an increased activity of the signature that we have identified in CTRB1 exon 6 deletion homozygous mutant mice, clearly indicating that what we are seeing in the mice is relevant to what's happening in humans. And we do pathway analysis of the full RNA-seq data sets in human, you can see increased activity of ribosomal biogenesis, regulation of transcription, ribosome assembly, ER stress, and topologically incorrect proteins, clearly indicating that the model that we have generated is relevant. And not only that, with Eduard Montaña, we have been able to identify a few organ donors and have access to tissue from a few organ donors who carry the variant. And this is expression of AGR2, which I told you was one of the most upregulated chaperones in the mutant mice, expression in wild-type pancreas, lack of detection of AGR2, and in we have looked, as of now, at 2 heterozygous individuals. In both, we find very marked upregulation of AGR2. Obviously, the question is, what happens with KRAS? This work is ongoing. We only have partial data. We've used various models and various protocols. We've used PTF1A-CRE, we've used PTF1A-CRE ERT, which we think is more relevant in conjunction with the exon 6 deletion mutant allele. In these conditions, as Mariano and Carmen have very nicely shown, it is necessary to induce an acute pancreatitis, otherwise, or induce other types of damage, otherwise no pancreas cancer develops. And we have used mice in basal conditions, or upon inducing acute pancreatitis or administering a high-fat diet. And what you can see is that the rate of tumor development appears to be with a trend of being increased in the exon 6 deletion mutant mice. The number of mice combining these diseases is still small, but the differences are not yet statistically significant. We think this is related to sample size. And this is just, again, showing you that at 6 months in the homozygous, and at 3 months in the homozygous mutant mice, we see massive replacement of the pancreas tissue by preneoplastic and neoplastic lesions. So what fostered all of this is to show you that the mouse models can be useful, but can we prevent it? And for this, we are taking advantage of the fact that there is a drug called TUDCA that has been approved for use in sclerosing cholangitis, and which is an ER stress reliever. What we have here in these mice is massively an ER stress phenotype. So we have started treating our mice with TUDCA because it's an FDA-approved drug, and this would be a drug that could be potentially used to prevent pancreatic cancer development in subjects at risk. And what we can see, this is a small experiment, pilot experiment that has been carried out in the lab. And what you can see is that when we administer TUDCA, we have reduced expression of BIP and CHOP, two classical ER stress marker, of a spliced XBP1, a hallmark of the UPR, of the unfolded protein response, and reduced expression of AGR2, which is the major chaperone upregulated in these mice. So we think that there is potential for applying this kind of strategy. It is complex, it's long-term, it's not going to be tomorrow, but the concept we think makes sense. So I hope I've convinced you that going from human to mouse and from mouse to humans is a valuable strategy. And I just want to finish by saying that in Spain, most of you who are here are Spaniards, but I just want to do a little bit of marketing of Alipanc, which is an alliance for research against pancreatic cancer in Spain. It includes clinicians, basic scientists, translational scientists, and we are very happy to work together in this alliance to beat the war against cancer, against pancreatic cancer. And this is the people who did the work. Let me see. Yeah, here you have Irene, who is in the audience, with Cristina, who did the last part of the CTRB1 project, Chengxi, Jaime and Mireia, who have been working on both projects or on the bioinformatics analysis, all the CNAO facilities who have been instrumental in doing the work that I have mentioned, and obviously this is inspired by our long-standing work with Nuria and our collaborators Miguel Lafarga, Nelson Ducetti, Scott Lowe, Fayaz Nora, and on the diabetes side with Jorge Ferrer and Eduardo Montaña. And thank you very much.
Thanks, Paco. Great talk. Do we have any questions from the audience? We have time for a quick one question before we move to the next speaker. If not, I will ask you, Paco. I think prevention, early detection is so important. Do we have any good preclinical models for IPMN and other types of precursor lesions that we can use in our studies?
Well, yeah, there are models for IPMNs. The GNAS-based models do yield more IPMNs than the ones. We are focusing on the acinar cell because the biology is so rich. But yeah, I think there's space. Actually, I think the space for IPMN is fantastic, not only in the mouse models, it's fantastic in the humans. I think that we should be doing clinical trials of prevention in patients with low-grade IPMNs, yeah, definitely.
And microbiome, you mentioned microbiome again. We need to be collecting these samples from our patients, from our high-risk individuals. Maybe they are sources for biomarkers for early detection, response to treatment.
Absolutely, and we need to do that ahead of time because otherwise we have reverse causation biases. So definitely, this is long-term projects, but very important because the war is formidable, as I told you at the beginning.
We need to go at all fronts. Any more questions we have? Oh, please, Bruno.
Thank you, Paco. As always, excellent talk. More of a curiosity. In our 5A2, it's interesting that when it's only 50% of the proteins expressed, it goes to regulating pro-inflammatory and not differentiation markers. And when you treated with antibiotics, did you see a switch in that regulation?
We have a problem with that. The only antibody that worked for CHIP PCR was discontinued. We're struggling. And we haven't been able, I mean, you know that we do a lot of ChIP-seq in the lab and a lot of cut and run, but if you don't have good antibodies, you can't trust the data. Therefore, I would think it would switch, yeah, but we haven't been able to do the right experiments. we're pursuing that. Very important question.
Okay, thank you. I think we don't have time for more questions, but thanks a lot, Paco. A great talk. And let's move on to our next speaker, Dr. Jen Morton. She's a group leader from CR UK Scotland Institute and also a professor at the University of Glasgow. So her research and her group are dedicated to understanding pancreatic cancer biology and new therapeutic strategies. And today she will focus on the role of the tumour microenvironment that drives chemoresistance, one of the key challenges in pancreatic cancer. So thank you, Jen. Thanks.
Thank you, and I'd like to thank the organisers for the invitation to be here today, all the way from sunny Scotland. So I'm going to dive straight into our mouse models, which is forward. Oh, cool. So everything I'll talk today, we use the KPC mouse model. We also have a load of other models in the lab, layering on different genetics that we find in the human. But for today's talk, I'm just concentrating on the workhorse KPC model, which I'm sure you're all familiar with, but I just wanted to point out that it is resistant to chemotherapy, which is important for a lot of our studies and a lot of the things I'm going to tell you. So mice get quite a bad rap, particularly from people that read maybe subcutaneous tumour manuscripts, etc. But I would say it's really not easy to cure cancer in the mice. We've tested maybe, probably up to 200 different concepts now in my lab. Less than 10% of them will have any effect on survival. Less than 5% even result in growth arrest. And now, more recently, we've seen about 3% overall result in regressions. They're also not that homogeneous. So we've done some studies looking at both inter and intra-tumour heterogeneity, taking multiple biopsies across mouse PDAC, and we find not only do they map closely with human PDAC, so this is a lot of RNA from the short read archive. This is actually, this is mouse PDAC mapping. These are human pancreas. This is gastric, actually. And when you take human data, actually, they also, if you replot more human data, they do this as well, which might be a cell of origin issue. But we can take multiple biopsies across different tumours and see that we get quite a lot of intra-tumour heterogeneity in our mice, including this real random one here, which is mapping. These are mapping with lung, but we definitely didn't accidentally put lung in the tube. So I'm going to tell you 3 different ways we've been trying to treat our mouse PDAC. The first started a long time ago, trying to target KRAS signaling before we had KRAS inhibitors, actually. This is our kind of preclinical trial methodology, where we just have a load of KPC mice. We wait for them to develop tumours. We palpate them every week. When we detect a possible tumour, they get ultrasounded and rolled onto treatment, and then followed by ultrasound imaging throughout the course of treatment. And this is what happened initially when we targeted both MEK and mTOR1/2. We got some quite nice extensions in survival in the mice, but what was more impressive really was that we got robust tumour regressions in all our mice, although you can see that a lot of them bounced back really quickly. This one didn't, and stayed responsive for quite a while, and we could restart treatment. I've gone backward. Eventually it became resistant and took off. But you can also see nice resolution of symptoms in the animals. So this is the massively dilated common bile duct when we put the mouse on treatment, and that completely resolves. So yeah, as I said, you see robust tumour regression in week 1, but it's actually pretty transient. And so we've started using the RAS inhibitors now. So this is with the Mirati KRAS G12D inhibitor. Again, we see really nice extensions in survival. We see nice tumour regressions, and they stay down for quite a while, but then bounce back up. And we see the same with the pan-RAS RevMed inhibitors. But we've been trying to work out why we get recurrence. So we do see this, oops. I'll try not to swear on the microphone. We see loss of tumour cells, which actually, sorry, this is not working, which is actually sustained until endpoint. So we see loss of tumour cells even at day 5, but that's still at endpoint. We see far fewer tumour cells, and we see increased fibrosis early on, which is maintained until endpoint. And we also see the same with KRAS G12D inhibition. We see that our target inhibition is sustained, so we see less phosphor even at endpoint. Not so much of an effect on the mTORC1 pathway. It's not a particularly good mTORC1 inhibitor. A nice effect on phospho-AKT. But what we find really interesting is that this marker that we use essentially to look at phospho-mTORC2 signalling, phosphorylated NDRG1, is normally expressed in the tumour cells, but when these tumours become resistant, it's no longer expressed in the tumour cells, but it is expressed in the calves, even though the tumours are still bathed in inhibitor. And this is specific to pancreas because we have some accidental models of cholangiocarcinoma, and we don't see the same thing there. And we did actually a pilot experiment to see if we could treat PANINs with the RAS inhibitors and also with the combo to see if we could use it as a preventative or very, very early intervention. I'm not showing the data, but unfortunately we could clear all PANINs, but then the mice recurred with PDAC just about as quick as regular KPC mice. But what we found really interesting is that in PANINs, actually, what we see is that this NDRG1 is phosphorylated in the calves in PANINs and not in the tumour cells. And it is, in that case, it is still responsive to treatment. So when we treat these mice, we see expression or phosphorylation go down. So we still don't understand the mechanism, but we're wondering if this signalling is also a way to promote PANIN survival and might be interesting in tumour initiation as well. And this, we shout out to Dieter, because this is the plan at the moment. We're actually going to manipulate the calves instead. I don't think I need to summarise this part. I didn't time myself. I'm going to switch gear a little bit. Well, initially we looked at this as a stromal target, but as the story goes on, you'll see that maybe we aren't targeting the stroma after all. We got interested in a protein called gremlin-1, which is a secreted BMP antagonist. It's overexpressed in stromal cells, it's upregulated in pancreatitis, it's both a marker and a driver of fibrosis, and high expression correlates with poorer survival in the patients. So we actually, there's been some conflicting studies reported on GREM1, but it's clearly doing something in pancreatic cancer. So we hooked up with a drug company who had an inhibitor of this protein, or an antagonistic antibody. And you can see here that combining with chemotherapy had a pretty minimal effect on survival, although technically statistically significant, but probably not biologically significant, because you can see that the tumours pretty much keep growing. But then, because of various studies in the work, and because this is before the days of KRAS inhibitor, when we started this, we decided we would combine with MEK inhibition, because it's been working historically much better as a backbone for testing targeted therapies than conventional chemo, at least in our hands, even though it does nothing on its own. So in that context, we got significant extension of survival in our mice, and some nice tumour regressions. And they stayed down for quite a long time, and we repeated that with a second MEK inhibitor and saw the same effect. So that resulted in a clinical trial, which is not perfect, but at least the patients will be going on the antibody plus MEK inhibition as maintenance, rather than waiting till progression. So the first patients are on trial in Glasgow, but we still don't really understand the mechanism for this. So we looked in the tumour microenvironment because originally we thought that we were targeting fibroblasts. But in the short term or at endpoint, the combination or the gremlin inhibitor alone had no effect on the alpha-SMA positive myofibroblasts. It had no effect on total fibroblasts, and it had no effect on collagen deposition. Oh, that's very blurry, sorry. We then resorted to geomics analysis, so we could try and tease apart the signals in the tumour cells versus the CAFs versus the immune cells. And what we found in the CAFs was extremely, well, I was going to say disappointing, but obviously splicing is super interesting. We should follow that up. But we didn't see any obvious, we saw very few genes, individual genes dysregulated. What came out again and again in the tumour cells was enrichment of genes repressed by RAS. And at that point, we went back and we looked in our single-cell RNA-seq data to see exactly all the different cell types that gremlin-1 is expressed in. And so you can see here that actually gremlin-1 is expressed in quite a lot of the epithelial, ductal, and acinar cells of these are tumour cells, but with an acinar or ductal flavour. And so we thought maybe in the tumour cells, actually, this biology was important. And so again, this is just bulk RNA-seq now, but we saw that proliferative signatures were upregulated by anti-gremlin, and that KRAS activation was upregulated, while KRAS deactivation was downregulated, similar to what we'd seen in our tumour cells from the geomics. So we're wondering now if anti-gremlin shifts the balance of tumour cells to a more proliferative phenotype via activation of KRAS signalling, and that's what makes them more susceptible to MEK inhibition. And now we want to go back and look and see if we would see even better efficacy with KRAS inhibition, and if that might influence also the de novo fibrosis that we see. So I showed you that anti-gremlin has limited efficacy in combination with chemo, but with MEK inhibition, it is effective against pancreatic cancer in terms of prolonged survival and tumour regression or long-term growth inhibition. No impact on CAF numbers and few transcriptomic changes, but it does shift the balance of the tumour cells to a more proliferative phenotype, and maybe enhancing susceptibility to MEK. So yeah, this work is ongoing with KRAS inhibition now, but we're optimistic. I still have time, right? 5? Okay. Okay, so just very briefly in the last 5 minutes, a completely different again, mechanism of resistance potentially in the tumour microenvironment, again this time. So we've been looking at radiotherapy in pancreatic cancer, which is controversial. I think the data show both pro and anti-tumour effects, and the data from clinical trials is not super promising. But there are, potentially effects on, or local regional effects and effects on recurrence, or downstaging tumours to make them potentially more operable. So we have this SARP platform in the Beatson where we can give targeted radiotherapy to our pancreatic mice. We can use contrast-enhanced imaging combined with ultrasound imaging, so we can actually find the tumour and target it quite well. So we have tumour planning or treatment planning software where you can target the tumour quite effectively, and you can see that we get nice induction of damage. Nice induction of damage in the tumour, but we spare the kidney, the liver. We get some intestinal effects, but the mice survive. The mice tolerate it quite well, and you can just see that the effects on the hemopoietic system are very minimal compared to total body irradiation. So if we just give radiotherapy alone, we see limited efficacy as a single agent, although there was a hint that there might be some benefit in smaller tumours. When we do hypofractionation, we can see that the efficacy improves a little bit with either 3 times 6 Gray in the yellow here, or 1 times 12 Gray. There were cells there. You're going to have to trust me that we see in short term in the microenvironment, we see a big influx of myeloid cells and lymphoid cells and a real reduction in the tumour cells. But in the long term, at 3 weeks post treatment, that's long term for a mouse, we see, well, we don't see anything, but I promise that we see a reversion back to the normal state. We don't see an increase in total CAFs or ECM, which is interesting because clinically fibrosis is reported following radiotherapy. All we saw was kind of qualitative changes in the extracellular matrix, which we're following up on. Interestingly, we also see increased metastasis in our mice, which we're also following up on, quite dramatically so actually, which might explain the failures in the clinic. We don't see a radical increase in lymphocyte infiltration. This is at endpoint, but what we do see continued to endpoint is increased tumour-associated macrophages, which is kind of dose-dependent. So we wanted to know if we could radiosensitize. Now, depleting all macrophages isn't necessarily the best way to go. We have done it in the past and seen some extensions in survival, but we've now got a mouse model where we can just prevent macrophage or monocyte infiltration into tumours because the mice lack chemokine receptors, CCR1, 2, 3, and 5. So you can prevent homing of the macrophages to the tumour, and this is super preliminary, but we're seeing some nice extended survivals now in those mice. So potentially, we think that in this case, the macrophages are playing, or the infiltrating monocytes are actually playing quite an important role in the recurrence and probably in the fibrosis as well. So just to summarise that, I showed you that the PDAC radiation alters the PDAC TMA. It can lead to tumour-associated macrophage accumulation. Targeting the TAMs might enhance sensitivity, but probably more likely we have to target both the monocyte and the macrophages that are already there, because we know that the TAMs can also support the adaptive immune response. So with that, I guess my overall summary is that pancreatic cancer is very complicated and difficult to treat even in the mice. But I want to thank Team Pancreas at the Beatson services, who are amazing and without whom we couldn't do all of our mouse work. The geomics guys in the University of Glasgow, all of my international collaborators who aren't on the slide, unfortunately, and the funders. And with that, I'm happy to take questions.
Okay, thanks, Jen. Great talk. Do we have any questions from the audience? If not, I'd like to... Oh, do we have one? Sorry, I was going to interrupt. Okay, sorry, I didn't...
You mentioned this striking increase in metastasis in the RT-treated mice. Have you, and you've mentioned some changes in the stroma, have you actually looked at the at phenotype switches in the tumor cells, or were there any changes in terms of basal versus classical phenotypes at the tumor cell level?
And they're all quite basal anyway. So we haven't seen any significant switches from. So sorry, Peter, we've only done basal versus classical, but we haven't seen anything significant.
And the metastases are to which organs?
Oh, so that's interesting, because we get a few more to liver, but we get many more to lung.
Thanks.
Do we have any more questions from the audience? If not, just a comment about recurrence, because we heard from Bruno this morning about these stem cell inhibitors. Maybe that could be something to look into.
Yeah. I know I need to go back and
Get his number before you go.
Look at the gene lists.
Okay, thanks, Jen. Thanks for your presentation. Thanks for the questions. And so let's move to our final speaker, Vasiliki Iliaki. She's a postdoc at the CNIO with Mariano Babacid and Carmen Guerra. She's received many awards for the work that she's done so far, one of our early stage researchers. And she's going to talk to us today about her recent work about combined targeted therapies that can induce complete regression of pancreatic cancer in mouse models. So thanks, Vicky. Whenever you're ready, thank you.
Indeed, I would first like to thank Mariano and Carmen for this opportunity today to represent our lab here today and to present some of our recent data. So we have seen quite some introductions so far in PDAC, but I will first just briefly mention that PDAC is a very RAS-dependent cancer, as 95% of all PDACs express mutations in KRAS, with the two most common mutations being the G12D and the G12V oncogenes. Now, our lab works on targeted therapies, and as Mariano mentioned before, it's a field that hasn't seen much advance over the past decade, as so far we still rely as first-line therapy on cytotoxic chemotherapy, and very few targeted therapies have been approved so far. So in the lab, what we do is we study the KRAS signaling network, and we selectively inhibit different signaling nodes in combination and separately in order to achieve tumor regression. We do this by using the genetically engineered mouse model, or GEM, that was generated in our lab by Carmen Guerra, where PDACs express the KRAS G12V mutation and are knocked out for the tumor suppressor P53. So in this line of research, our group published in 2019 a study where genetic elimination of EGFR, the upstream signaling node of KRAS, and RAF1, the downstream signaling node of KRAS, led to complete elimination of the tumors that were used in the study in half of them. The other half showed resistances. We should mention here that the initial tumor sizes that the combined elimination took place was quite small. So the tumors were 20 to 25 cubic millimeter maximum size and lower. We then continued in this approach by using bigger tumor sizes. So what we observe is that when this combined elimination of EGFR and RAF1 starts in bigger tumor sizes, the majority of the tumors are resistant. Oops. No, sorry, something is wrong. Here. So this indicated that we had to study the resistant mechanism. As we saw, the majority were resistant tumors and bigger tumor sizes. We first started by studying the mechanism in vitro. So we started using PDAC cells and that do not express EGFR and RAF1, so they're knockout for these two genes. And we used transcriptomic analysis, RNA sequences, to detect the upregulated signaling pathways, where we observed increased activity of STAT3 together with other inflammatory and basal-related pathways. Next, we wanted to validate this increased expression of the phosphorylated form of STAT3. So STAT3 gets activated when it's phosphorylated in the tyrosine 705 in this residue, and what we observed was in vivo in the GEM model, when we have effective elimination of EGFR and RAF1, we observed increased phosphorylated STAT3 in immunohistochemistry in PDACs. So after we identified our third signaling node as STAT3, we moved on to functionally validate its therapeutic efficacy. So we first eliminated in vitro in PDAC cells EGFR and RAF1 as they are the flox genes with the use of adenocriviral particles, and then we added shRNAs to target to inhibit the expression of STAT3. We saw complete cell death. However, we did not see any toxic effects by just using the shRNAs here, only by infection with those shRNAs. This also validated that STAT3 alone is not a therapeutic target for PDAC. So after defining the three signaling nodes that need to be targeted at the same time, we moved on to the combined genetic elimination. So what we did is to create a new GEM model in a lab, a new GEM strain, where we use the three floxed alleles, EGFR, RAF1, and STAT3, and PDAC cells also express CREAT2 under the promoter of ROSA26. We let tumors grow in this new GEM model. We sacrificed the mice at human endpoint, and we generated many mouse PDAC cell lines that we then implanted in the pancreas orthotopically of black 6 isogenic mice. So we let the tumors grow, and when tumors reach tumor sizes from 40 to 100 cubic millimeters, we subjected the mice to tamoxifen diet. So tamoxifen diet induces the expression of recombinase CRE that then recombinates and deletes the three flox alleles simultaneously. What we observed, to our surprise, there were quite encouraging results, is that in all of the mice and all of the tumors, we saw complete tumor regression for up to 300 days post-implantation. So we should mention here that we used the tamoxifen diet for around 30 days to achieve the regression, and then we removed tamoxifen diet and we replaced it with regular diet, and we did not observe any tumor relapse. Here we used in total 36 mice. So we implanted 12 different cell lines in triplicates, and these cell lines have been previously validated as resistant to the double combination, to the double elimination of EGFR and RAF1. So after this proof of concept that elimination of these three targets works, we wanted to make our results a little bit more appropriate for the clinic, so we started with our pharmacological validations. Unfortunately, up to recently, but still, there are not any RAF1 selective inhibitors that we could use. However, we have seen, as we saw in the previous talk, recently the big breakthrough of all the RAS developed inhibitors. So we have a big variety now coming up, also mainly pan-RAS inhibitors and also selective inhibitors for the oncogene of the mutation of G12D. In our case, we chose to use the RMC6236 or daraxonrasib as it's a pan-RAS inhibitor because our model expresses the G12V mutation. Now later on we will have some talks from clinicians that they can tell us how RMC6236 and other inhibitors are doing in early clinical phases. However, just to briefly mention, it's it's a big breakthrough, so very promising data. These inhibitors, they extend survival, however, we still see resistances in patients. So our main goal here was to go for the resistances, to target and see if our approach in our models would develop resistances or not. Again, we implemented this drug, this inhibitor, to our triple strategy. So for the inhibition of VEGFR, we used afatinib, that is a pan-ERBB inhibitor, in order to inhibit both the activity of VEGFR and ERBB. And for STAT3, we used a selective degrader. It's a product that was published in 2019. It's called SD36. So we started with the orthotopic models. We implanted our KPF cells to the pancreas orthotopically of isogenic black 6 mice. We let the tumors grow of tumor sizes higher than 100, equal to 100 or higher cubic millimeters. And we started with our triple combination. We saw, again, similar to the previous results, a complete regression for up to 200 days post-implantation. Here, we repeated the same approach. We treated the mice for about 30 to 40 days, and then we removed the treatment to see if we would have any relapses, but the tumors didn't come back. Monotherapy with the RMC6236 inhibitor had an effect. It did not induce any tumor regression, but extended the tumor growth for up to around 3 weeks. Here I would like to say that we have performed all the double arm studies, trials. So we have tried all the possible combinations by 2, and we have not seen any tumor regression. What we see is significant delay in the tumor growth for from 2 to 4 weeks, depending on the combination. And as it was said earlier in the previous talk, the ultimate challenge here is the GEM tumors. So we used our mouse model that we know is far more complex and more difficult to cure. So first we tried monotherapy. We tried the effect of the RMC6236 or daraxonrasib. And what we saw was some variability in the response with significant delay in the tumor growth. We then implied our triple combination of RMC, afatinib, and SD36, the product against STAT3, and we treated tumors of higher than initial tumor size than 100 cubic millimeters. What we have seen so far is no resistances. In most of the cases, we see complete tumor regression for up to 200 days in some mice treatment. And in almost the cases, we see tumor regression, most of them complete, but most importantly, we don't see appearance of resistances, which was basically what we were aiming for. So, of course, this data we were quite excited, but we couldn't, we should continue by implementing in our studies human patient-derived samples. So, we used PDX cells that we implanted in the pancreas of immunocompromised mice orthotopically. So when the tumors grew, we used our triple therapy, triple combination of daraxonrasib, afatinib, and the product against STAT3, SD36, and we observed, again, complete tumor regression. Here, what was quite promising is that these samples, they express a big variety of mutations, and they have almost all driver genes, the usual 4 driver genes, mutated. In this case, the 3 PDX models that we were used, they express the G12D mutation. So we also included experiments where we used the MRTX1133 selective inhibitor for the G12D mutation. We had a similar result, And now we have also included in our studies not only cells, but also tumor pieces and other samples that they also express different KRAS mutations. The results are similar, so we see complete regressions without any resistances. One of the most important things that we were actually, we wanted to achieve is to, and also is also the main goal of the lab, to achieve tumor regression without any toxicities. So far, in our experiments, both immunocompetent and immunocompromised mice, we have not seen any toxicities, or to say it better, we see high tolerability of this treatment. Both, we don't see any loss of the body weight, and we see all the organs have intact structure. And we've also performed some blood analysis, checking the blood counts and the metabolic parameters that seem to remain unaltered. Now, what we have seen so far is that basically we defined a triple strategy targeting 3 signaling nodes, EGFR and ERBB2. At first RAF1, so now pharmacologically RAS and STAT3. We have included pharmacologically in our studies afatinib, daraxonrasib or RMC6236, and the selective PROTAC SD36. One of the most important things is that we have included a big variety of experimental models. So from GEMS to orthotopic allografts, xenografts, both cells and tumor pieces. And we have also included a lot of studies with patient-derived organoids with a big panel of mutations, KRAS and the other driver genes. Now, we were quite excited about our work. However, we know that we have still a long way to go for the clinic. So now our current research trials include refining a bit this therapy in order to avoid future toxicities in patients. So first, we know that afatinib is a very potent inhibitor and has been linked to some toxicities in the clinic. So now we're trying to replace it with other EGFR degraders or antibody drug conjugates that are more selective, so are expected to have much less toxicities in the clinic, especially in the combination with the daraxonrasib. We're also including other KRAS inhibitors, more selective for the G12D mutation, that is the most common in patients. And we are also now working in close collaboration with a small biotech company to optimize the pharmacokinetic activities of SD36, because right now is not optimal for clinical use. But we are also at the same time using in our studies other inhibitors, STAT3 inhibitors or another STAT3 products that have entered clinical trials. So we still are very confident that we can move this forward, and we're hoping to contribute more to the clinical scenario. With this, I would like to thank my 2 supervisors, Carmen and Mariano. I would also like to thank all the members of the experimental oncology group, but mainly Sara Barambana here, that has worked with me, no, this one, that has worked with me in this story. All of our collaborators that participate in our PDAC projects, and all the funding agencies. I would personally like to say a big thank you to the CRIS Foundation, because they have been long supporting our PDAC research and of course this project. So thank you for your attention.
Okay, thanks Vicky, great talk, really impressive results there with complete regression. Great to see KRAS inhibitors working, G12D. Do we have any questions from the floor? Yeah, we have 2 here. Maybe ladies first, yeah?
Sorry, my question is really a curiosity one. Since you haven't seen much toxicity in your mouse models with drugs that in the clinic instead are giving some problems, how are you going to compare that with the new drugs that you are testing, since you don't have a toxicity to compare it to?
Yeah, this is true. This is something that Mariano has been saying quite long for years, is that sometimes mice are more, they can handle more higher doses, they can handle trametinib that shows toxicities. So that's why we won't be preventive So that's why we want to see, we know that this is not toxic, but we expect it to be quite potent in the clinic. That's why we want to tone it down in the efficacy, and that's why we want to replace afatinib, taking into consideration that right now it's not toxic in mice, but would be expected in the clinic, mainly for the combination of RAS and EGFR, for the rashes that now they're currently, I think, already seeing in the clinic. So we are under discussion now to see how we can tone this therapy to avoid further toxicities in humans, yes.
That was excellent. I love this presentation. Couple of questions. One is with the genetic ablation, I think I saw you started treatment when the mice were around 100 cubic millimeters. Have you tried that when they grow more than that? Like if they reach 300, do you still see response with the genetic ablation? And then with the pharmacologic ablation, have you gone higher than the 100 millimeter to start the experiment? And the third question, sorry, is have you been able to see with a STAT3 inhibitor similar results to what you see with the degrader? Like, do you need to eliminate the protein, or it's an inhibitor going to work?
Yes. So the first question about the genetic elimination, just to clarify, I think I didn't clarify it in the presentation. Since this is an orthotopic model, first, the elimination happens only to the tumor cells that are implanted. And we have at first, when we're doing our experiments, since we included a lot of mice, we were also going higher in tumor size. The problem is that the higher the tumor size, the elimination is not that good. So it was a technical problem with the tamoxifen treatment. Mainly due to the speed that these tumors were growing, because if it's an orthotopic, so if you pass 200 cubic millimeters, then the tumors grow too fast and the tamoxifen elimination was not as good. But we had tumors of around 200 cubic millimeters that they was quite promising the elimination. So some of them were regressing. But when we sacrificed the mice, it wasn't optimal, the elimination. The pharmacological combination, we have, so the GEMs, I think they are also included in this graph, we have gone up to 300 cubic millimeters, and it seems that it's working. I think the curves are here, so we have at least 3 mice going higher. I don't know if trying around 400 cubic millimeters or 5, because it's nearly human endpoint, would be that appropriate in this model, but 300 cubic millimeters works so far. And the last one, we were asked this question, so that's why we're now trying this inhibitor, TTI 101, that is an inhibitor of the phosphorylation. So far, we have started the trial, so we don't know yet if we'll see. We're just setting it up.
Okay, thanks. One more question here.
Great work, congratulations. I was curious to see if you have looked at the tumor microenvironment in your triple treatment where you don't get resistance, and maybe compare it to the double treatments where you still get some effect, but you get resistance.
Yeah, so now we are in the process of characterizing our GEM model. What we see in the triple combination is that when the tumors regress, and they disappear, the stroma also disappears. So at the end of the treatment, when we see 0 tumor, some of the mice have been sacrificed. When we see 0 tumor presence, there is no stroma, so the stroma disappears. In intermediate steps, we are now checking how, while the tumor regresses, how the stroma behaves. We haven't finished this analysis. What we know for sure is both in the orthotopic models and the GEM, when we make the tumor disappear, there is no stroma, so because we have seen the sections of the pancreas. Now, the double combinations, now we're starting to characterize how the tumors that finally grow are modified, their tumor microenvironment, stromal and immune compartments. We are ongoing, we have the characterization.
Thank you.
Okay, thanks. I think with that we can close the session. Thanks, Vicky. Thanks, Jen. Thanks, Paco, for your talks. We've seen how mouse models are so important in our research and in our fight against pancreatic cancer. Mariano, I don't know if you want to say a few words when we close the session, or we just go straight for lunch. Okay. Hopefully we'll have food this way, I think. So enjoy the rest of the symposium, and thank you, everybody.